our website, the globalimmunotalks.org. In this website, you will be able to see how we started this initiative because we truly believe and we work towards immunology without borders. The motivation for this initiative happened at the time of COVID. We realized we needed to stay connected. We realized we couldn't go to our regular places of work, but internet could allow us to be connected. And so at that time, in early 2020, we funded this Global Immunotalks initiative. There are two main fundamental goals, I would say, about this initiative. First is to inspire immunologists across the world in an egalitarian manner. We realize that we don't, all of us, have the same access to the latest of immunology. We wanted to contribute to making that access more readily to anybody having an internet connection. Obviously, at the time during COVID, we couldn't travel, but that is not the only time in which we cannot travel. There could be many circumstances that make it challenging to go to meetings, to travel and go to listen to these speakers. So we wanted to bring really immunology without the need of traveling everywhere in the world. So how did we started this? This was an initiative between Elena and myself, but I really want to recognize Elena. This is a younger picture for both of us. Elena is a professor at the University of California in San Diego. As you can see, I'm in the process of moving, so we have to update my entry here. But Elena and I are close colleagues, and we both trained in Argentina, so we know firsthand the wonderful opportunities that we have across the world for training, but also the fact that sometimes we don't have access to the same speakers, the same opportunities for learning. So Elena and I started this initiative. We recognized early on that we needed to have organizers across the world. We have had a series of organizers. This initiative is now a 5-year-long initiative. Our initial organizers, you might recognize many of them. They are very, premier immunologists. They do their immunology science across the world. So we realized we needed to have colleagues from everywhere. Now, these are our previous organizers, which form part of our board, and I would like to introduce our current organizers to give you a sense of how international this effort is. You can see in this poster colleagues such as Asia Rose from India, Hai Chi from China, Gabriel Rabinovich from Argentina and now Spain, Mira Merat from the US, Bali Pulendran from the US, Ana Maria Lenon from France, and Caetano Reyes Sousa from England. We definitely wanted an international coverage. You will notice it's not, of course, everybody, and is not representing yet Africa, something that we definitely would love to expand on. Now, let me tell you about the talks, and let me introduce you to where you could find the talks. You can find them here by ear, and I would like to share with you one talk. We had one of our colleagues that does research in Africa, but I have to admit, we do not have enough representation from Africa, and I hope you can help me make sure and help all of us in Global Immunotalks to change that. Sorry that I'm scrolling, but here you can see many of the talks that we have in 2020. I would like to introduce you how you can access these talks. Let me do this by the example of Sasa's talk. You can see this was early in 2020. So we have talks until yesterday. No, today's Friday. Until 2 days ago, we go live every Wednesdays. But what you can see is you can go to all our talks. If you click on the title, you will be taken to YouTube. You can see here, for instance, you have Sasa's talk, and there also you can see comments on how well it's received. You can see this talk has been viewed more than 1,000 times. Now, obviously, we want to do better at this. I want to share with you other aspects of the global immunotalks. We realize that these are many talks. We have more than 200 talks. So we would like to create what is a living textbook, if you wish, assembling the talks into collections. If any of you happens to be interested in cancer immunology, you can go to our collection site. This is a cancer immunology collection. You can have a list of all the talks that relate to cancer immunology. We are working on a collection on infectious diseases. We are working on a collection on mucosal immunology. And hopefully we will bring more collections that are thematically arranged. Now, how do you access this? Again, this is for free. This is our next speaker next week. You can go to the website, but this is where it's important. You can join us. This is a free registration. If you register, you get a Zoom link, okay? Now, you don't have to register if you're busy. You can just directly go to our Global Immunotalks channel, and you can directly. So sorry. I apologize. There is like the volume is on, but you can go directly to the YouTube Global Immunotalks channel. We can play it again. I guess here. Let's see. I don't know. Okay. You can go to the YouTube Global Immunotalks channel and directly see without registering. If you register, you get an email, and then you remember and you get all the information about the upcoming talks. I want to share with you some of our statistics. We have had over, I'm hoping you can see this information here. We have had more than 700,000 views. This is exciting. However, we always want to really achieve our goal. And as you can see, if we break down these views per countries, the vast majority is still the United States. We have viewers from many other countries, but we still have not reached your region. And so I'm incredibly grateful for this invitation to present our work and to make this incredible collection of talks, an ongoing collection of talks and live talks that you can watch and allow you to learn more immunology. I want to do one last comment, and then I'll open it for questions. Again, if you go to our website, I want to introduce you to a sister initiative that I'm very excited about, and that hopefully we can also bring it closer to you. So along with the Global Immunotalks, we started Global Immuno courses. We've done this for 2 years, and this is going to be the 3rd year where we brought this fundamental course in immunology to India. We fundraise, we make sure everybody that comes to the course doesn't have to spend 1 rupee in India, whatever denomination of the money would be in each of the countries, and attend a fundamental, excellent course in immunology. If you go to the website, I invite you to see a summary of what these courses have provided. You can see me there teaching here in India, fundamentals of immunology. We have wonderful forums where we connect with colleagues across the world. So here we go in person to the site, teach fundamental immunology, but still connect with outstanding speakers across the world. You can see here how we connected, for instance, with Professor Ann Rostin at UMass in our first course in India. And we have expanded our instructors. You can see from our 2024 course also in India. We have had outstanding instructors such as Professor Sarah Ghosh and Professor Laura McKay, who joined me in teaching these fundamentals of immunology. So with this, I guess at this point, I'd like to stop sharing and address any questions you might have. I hope you find this of interest. We have a Twitter account, or an X account, we have a Blue Sky account. If you're interested, please join us. We look forward to your feedback. We hope we can have organizers, speakers, and viewers from your region. And we hope we can bring the latest of immunology really to anybody that can just have a phone or a computer and an internet connection. Thank you for everything you do, and let me know any questions you might have. Thank you for having me. I would really appreciate your questions. I see that the microphone, I think, is going around, and any suggestions. We want to make sure this is something good for our global community.
Thank you, Carla. Thank you. We really appreciate a very insightful talk. And I think we can invite a few questions, but maybe before we do that, let's clap for Carla for... Yes. Good. We can take two rapid questions.
Okay, thank you very much. I'm known by the names Dennis Omara from Uganda Virus Research Institute. Thank you for that presentation. I would like to know to what extent these immunotalks helped students in the line of mentorship, especially the ones that are always going for postgraduate training. Yeah, thank you.
Dennis, right? Thank you so much. That's an excellent talk. Excellent question, sorry. So, sorry, it's a little bit early here. This is part of what we hope the global immunotalks can impact. Let me tell you the feedback that we have received about these talks. So, in terms of opportunities for learning and mentorship, we have learned two aspects that I think might be of interest and might be useful also for you and your colleagues. One is that these talks, because they are a collection of outstanding latest, developments in immunology, have been extensively used in the context of journal clubs and advanced courses that are taught in graduate school. So I have learned from many of my colleagues, sometimes you want to have like a focused seminar series, but you cannot bring all the speakers, right? That is costly, they're busy, they are very far away. But what professors have done is they can select some of these talks and use them for journal clubs. So one possibility is you can identify a paper for one of the speakers. The students read the papers and then you listen to the talk, and that provides a great opportunity for learning. So that is for learning. The second, more focused to your question, Dennis, about opportunities for mentorship, I have learned that many of my young colleagues have used this seminar series to identify potential mentors. This is a great platform where you can listen to a talk from somebody you're interested in working with or in having as a mentor. This can give you an opportunity to listen to their research and then contact them and have an idea of their approaches to science, obviously their details on their research. And I have learned that many graduate students that are finishing their PhD have used this platform to identify postdoc mentors. I hope this somehow addresses your question. I'm sure we can do better, and I'm looking forward to hearing from suggestions you might have. Please feel free to email Elena or myself or any of the organizers.
Thank you so much, Carla. Nice to see you online. This is Lucy. My
Lucy,
question.
I can hear your voice.
Okay, great. Thank you. So my question is, I know you also serve under iOS now. So I'm just wondering, just for the audience to understand, how does the iOS together with now the ImmuTalks and what you're doing link up? Because I know iOS
Thank you
also
so
organizes
much.
other courses. Thank you.
Thank you, Lucy, so much for yes, introducing that aspect of like my commitment to IUIS and my current, involvement with IUIS. So I hope everybody is familiar with IUIS, our International Union of Immunology Societies. This is the society that brings all of us together. I have personally been, involved and contributed ideas, effort, time to IOS over the years, particularly with courses that IOS organizes across the world, similar to the Global Immuno courses, something that we want to do also as we move forward. I am currently the president-elect of IUIS, and I hope we can work with all of you to really bring the mission of IOS, which is immunology without borders. I think that initiatives like this that make excellent science accessible for free to anybody across the world that has, an internet connection is one step towards that. But unless we know each other, unless we're familiar with this, unless we also build, I'm thinking, Dennis, we could also build local groups where we can come together to see these talks, to discuss with one another, to attend the courses, to learn with one another. Unless we combine this opportunity with local efforts, right? I think we don't make the link between this opportunity and the effective, impact in each of the regions. So as president-elect of IUIS and with our current president, Rita Caserti, I hope, and with all, we have wonderful people, Lucy also, of course, is very much involved, and many of you, we hope we can bridge those efforts. I think we need local groups, local communities that come together to build that critical mass, that take advantage of opportunities for learning like this one. And I'm hoping we can make this a reality. I think this opportunity to connect with you today is one example of those bridges that we can bring and make this a reality. Lucy, I don't know if you agree with my perspective, but I really thank you for this invitation, because I think if we know each other, if we know what we can do together, and if we build, effort everywhere across the world, that's what really will make immunology without borders.
That's excellent. Thank you so much. And it's just great for you to share your plans and future for immunology. Thank you so much.
Thank you, Lucy.
Okay, let's have the last question here.
Yes.
Thank you very much for this very good initiative. Just wondering what informed the choice of the members for the advisory board. I can see, yes, from this region, we are not really very much into it. But maybe you can tell us what you considered in that recognition for the members in the advisory board. Number two, and this is from a question that has been asked, I think it's from a student. Do you normally have talks from research, that is publications, or you even share textbook literature, or what is the scope? Thank you.
Thank you, and I'm sorry that I didn't make that clear. Let me start addressing your second question, because that's the simplest one, and I would really like your help for the first one. So the talks, these are basic, translational as well, science in immunology, delivered by mostly established investigators. We also have some slots where we invite mid-career investigators, okay? So this is the latest in immunology. I think you bring a very, very important point. And to be honest, we haven't had the bandwidth to develop talks that are based on textbook. However, I think we could innovate with our global immunocourse effort, because that sister initiative is really more centered on textbook, but also sharing our knowledge as educators that run basic science labs. So we bridge the textbook with the latest as well. Okay, so the talks are advanced talks in immunology. The global immunocourse is a fundamental course in immunology, and I think your idea would be great if you could complement that with talks that would be based on fundamentals. We are missing that part. How did we choose? Let me go to your first talk. How did we choose the organizers? The first year, Elena and I started this initiative. We designed it, we invited the speakers, and we realized very early on that this could not be just Carla's and Elena's seminar series. Obviously, if you take two individuals from this more than 7 billion that we are in the world, we are limited in our knowledge. We are focused only in a particular area of science, of immunology. So we realized early on we needed to have colleagues that would represent, regions across the world. So we invited these colleagues. And of course, I have to admit that has been limited also by our knowledge. And that's why I'm so excited to be here with you today, because, I have met Lucy more recently, we met Sasa, but I personally have not had the opportunity to meet many of my colleagues in your region. And I believe, I won't speak for Elena, but I think that's also the case. So we really want to correct that. And the only way I feel to correct us is first to acknowledge it. We know we don't have that representation. And hopefully to open this opportunity to you and to interact with all of you to have representation, obviously from the whole continent of Africa. And I hope you accept this as an invitation that we need to change this, and that this limitation is just my personal limitation. And I think Elena's, as well. I won't talk for her, but I can talk for me. It's my personal limitation.
Thank you. Just follow up on how do you support this initiative? Do you have financials, or how do you make your money?
Yeah, we have wonderful donors. You can see this on the website. It's very limited amount of donors, and it's not a very expensive, you would say. I don't know if that's the right word, initiative. So we raise some funds, but this just supports some minor administrative cost to run the Zoom and to put things in the website. So this can be done with very, very limited, I would say, resources. We're very grateful for our donors, and we're very grateful for the speakers that absolutely donate their time and effort.
Thank you very much. Let's really appreciate.
That's what makes this for free,
Yeah, sure. Thank you very much.
Thank you for having me. I hope I need your help, like we all need your help to make sure that we have organizers, speakers and viewers that really expand our global initiative and make it truly global. I would say at this point, we are not, the name is an aspirational name. We are not truly global. We hope we can make that happen.
Sure. Let's work together on this.
Thank you so much. I look forward to connecting with you all.
Super nice. Thank you, Carla.
And thank you, Lucy, again for inviting me.
Yeah. And we hope you can stay with us as we get into the next talks.
Okay. Thank you.
Yeah. So it's my pleasure to introduce our next speaker, Professor Evans Egwim, who is an academic and researcher in industrial biochemistry and bioprocess, affiliated with the Federal University of Technology, Minna, Nigeria. He earned his first degree from University of Lagos and advanced his studies with a master's and a PhD from the University of Ilorin. Currently, he leads the Product Research Unit at the Center for Mycotoxin and Food Security, where he has developed innovative products and secured numerous patents, significantly advancing the fields of biochemistry and bioprocessing. Egwim has held several key administrative roles, including head of the biochemistry department, sub-dean of the postgraduate school, and director of multiple institutes focused on bioexploration and biotechnology. As the founder of the African Bioresource Network, he also consults for various companies across Africa, applying his expertise to enhance industrial biochemistry applications, and his academic contributions are substantial with 160 peer-reviewed publications, and he has supervised about 27 PhD students, 63 master's students, and over 200 undergraduates, reflecting his dedication to mentoring emerging scientists. Professor Egwim's leadership and innovative work position him as a key figure in the advancement of biochemistry and food security in Africa. Ladies and gentlemen, join me in welcoming Professor Egwim for his talk. Let's give him a round of applause.
Thank you so much. I'm very excited to be standing out here today to talk about the topic we have today. I will be changing the narratives from classical medical biotechnology to food sciences. I'm sure that will not distract you so much. Currently, I lead the bioprocess lab in the African Center for Excellence in Mycotoxin and Food Safety at my university, Federal University of Technology, Mina. So today, I'll be talking on the role of machine learning or AI and microfluidics in African fermented food to establish the full potential of African fermented foods as we look at the okay. All right, so we'll be looking at harnessing AI and microfluidics to unlock the health potentials of African fermented foods. Now, one thing we have in common here as Africans is that there are several staple foods that are made from fermented foods. If you look at the samples we have there, nearly every community in Africa have specialized fermented foods. And our interest is to bridge the gap or to bring together traditional fermentation wisdom with cutting-edge biotechnology to transform the African indigenous food into globally recognized functional foods and nutraceuticals. That's the underlining words, functional foods and nutraceuticals. Today, supplements, different kinds of supplements are either made from natural products or they are made from synthetic products, but we are now thinking about making functional foods from fermented foods. All right. So here we have different types of fermented foods. You go from milk products to cereals to sorghum to cereals. Sorghum. We have tubers, and then we have legumes. All sorts of these give us our different kinds of fermented foods, and they are all scattered everywhere in Africa. Now, the first thing that comes to mind when you talk about fermented foods are the microbial profiles. And the leading microbial profiles in these are normally the lactobacilluses. But the good thing is that there are several of these microorganisms. The lactobacilluses and the yeast lead the profiles. But after fermentation, one thing is clear. digestibility becomes increased, anti-nutrient, nutritional factors are removed, and then we have lots of availability of essential minerals like iron, calcium, and zinc. These are the things we know about fermented products. So if you look at this, for instance, you can go from the stuff like tubers, you have cassava yam, cocoyam, you can go on to have your gari, fufu, and all that you know. Then you go to melon, castor seed, you have your iru and ogiri. These are all condiments, food condiments, but the processes of fermentation goes on. Then you have the classical bacteria, microorganisms involved in this. Now, wow. All right. So the next thing we want to look at is generally, why is it that fermented foods are catching interest? The benefits of... Okay, so now, the first thing you want to look at when you look at the benefits of fermented foods are their nutritional enrichment. There's increase in mineral bioavailability, and then vitamins, and on. These are one challenges, one good things you can get from fermented food. Then the next is food security, where fermentation increases shelf life and preserves tropical foods. All right. So the next is the economic impact. Over 30 million rural households across sub-Saharan Africa depends on fermented food enterprises. So in these three aspects, we see that in the first place, fermented foods are actually giving us some good benefits. But we need to go beyond the general benefits of this. Why is it hanging? It's not giving us what we think of. Now, the first thing is to get bioactive peptides from these fermented foods. The principle is this: when you ferment protein-rich materials, with time, the type of organism, the conditions of fermentation breaks down the complex carbohydrate protein to smaller units. It is these smaller units that actually gives us the functional foods, or what we can use as our nutraceuticals. So here you have smaller units of the peptides that can act as enzyme inhibitors that can inhibit different kinds of other macromolecules like the ACE inhibitors, which are targeted for hypertension. So here we have seen on paper that from Bambara nut hydrolysates, you can have inhibition of 59% renin and a strong inhibition against ACE, which are well known to control high blood pressure. Now, we want to look at them. Wow. Okay, so the other benefits we look at is good. So you look at antihypertensive fractions or peptides, which can inhibit ACE to reduce blood pressure. We look at immunoregulatory cytokines, which can be gotten from biopeptides. And then we look at antioxidants from bioactive peptides. And then we also see anti-diabetic peptides that can be also used to regulate blood sugars. These are different bioactive peptides that you can get from your fermented products. Now, the question we have, and the big problem is, when you do fermentation, Several fermentation protocols will give you different fermentation time, different fermentation organism, different conditions. And in it, you have thousands of bioactive peptides. Now, if we go through our conventional means of recovering, identifying, and characterizing these biopeptides using your HPLC and all the conventional big equipment, it will take you weeks and months. And that is where our interest in using the microfluidics and the machine learning comes in right now. So in purification, for instance, where you want to target an ACE bioactive peptide, you have to go through lots and lots of purification, elimination, and characterization. But now, what we are thinking about, or what we are working on currently, is to introduce machine learning and microfluidic techniques so that we can pinpoint exactly what biopeptide of interest at any time and go straight to exactly what we are interested in. So now, what we do is that when you introduce artificial intelligence or machine learning, you can exactly predict what bioactive peptide of interest from any substrate at any time. We have done this, and then you can have as much as 99%, 97%, and all that accuracy, precision, and score. So now what we are now doing, our experience in our laboratory, what we have done is we have done some machine learning algorithm where we have different inputs. You can have your inputs, can be cow milk, goat milk, legume, whatever. You decide your input. Then you decide the microorganism of interest. Then you decide the inoculum size, the temperature, the time, the pH, and the steering time. These are different conditions that will give you different bioactive peptides. Then we use different model architectures like random forest with multi-output regression. And these are all the various models we have used to train our machine, to develop our models. Then you can have predictions. If you have the inputs, you have done the training, these are the possible inputs, outputs we are expecting. You can have the biological activity. For instance, you want to predict for ACE inhibition, you want to predict antioxidants, you want to predict antimicrobial, whichever of your interest. With your prediction, with your model, you can decide which of these are your interest. You can also predict the bioactive peptide sequence. This is the biggest challenge when you use conventional methods. The smaller the bioactive peptide, the bioactive molecule, the more difficult it is to isolate using the conventional method. So, but with this, I can know what factors will give me what. And so that becomes easy. When I know that, it becomes easy for me to go for bulk fermentation and get my bioactive peptides quick and early enough. Now, these are the predictions. When you do your input, you see the differences. Then you see the source cow milk. This is just an example. You have your lactobacillus, you have your time for 24 hours, pH this, inoculum size, and temperature. These are the inputs. Then you will now get your predictions from this, and it becomes easy. Wow. Good. So the predictive bioactive peptides, so you can now see the different short, very short bioactive peptides. You can see the first peptide there, 100%. The unspecified, 91%. Those are the ones that we have not gotten the identity. We have others, and they are all clearly predictable. Now, what that means for us is that you can also predict the bioactivity. Now, we can easily decide the desired peptide sequence and their function. This is where working towards a product becomes very easy for us. Now we can decide to make supplements from bioactive peptides. We can start to make nutraceuticals, which will not take you years to get into the market. I'm emphasizing this area because most of our young researchers here today, when we look at the big time sciences that we have looked at in this conference and at the workshop, you are imagining, how can that come to my level? The level is that for every one of you, you have a fermented product in your localities. They are very simple. The same principles we are using in infectious diseases, in immunology and all that, bring them back into your own community. Then you can get functional foods that are very, very available and can get to the market within a very short time. In my laboratory, every research is translated into the market at one level or the other. And that's what we are working on right now. So now, here was Professor Aman in my university establishing the microfluidic research unit. Here now he has signed an agreement with my university so that most of our work, he can now design the microfluidics from his engineering laboratory, and we can work on it to get the various kinds of bioactive peptides of interest. For now, we are working on the machine learning, and once we get all the machine learning, the models of interest, then we can go to the microfluidics. Now, for microfluidics, particularly lab on a chip system, it has helped us to minimize several working hours. Out of several thousands of biopeptides that you can see at once, we can now, with this, narrow down to the exact separation of interest using the various microfluidic techniques that we have known already. So we can now sieve according to their sizes, according to their charges, according to their migration capacities, and according to their other properties. So with that, with our AI machine learning techniques, we identify what we want, and with that, we can use the microfluidics to reduce both time and characterization, and it becomes a very simple thing for us to do. Now, at this level, with our interests and what is going on, it is important to know that multi-operations of a single chip increases throughput exponentially. So with that, we can extract as many as we want, and at what specific areas of interest. That makes it easy for us. Well, good. Then precision control. We can exactly know what we want at what time. That's very, very important, particularly as we work towards translational research. Now, double-based microfluidic screening achieves tens of thousands of droplets. Here, we are shortening time to from weeks to short hours with at least 90% correlation with existing mass spectroscopy validation. So this makes it very, very clear that we can achieve our purpose with the machine learning and microfluidics, and with that, we can have in control. Now, synergistic framework. With AI microfluidics, we are on our way to achieving high throughput target. Now, but at this point, we have gotten collaborations with microfluidics. We also invite others who may have other forms of models or other forms of predictive algorithms that can help us go further. We can go beyond functional foods and all that to other forms of omics, so that we can now be able to predict the mechanism of action. We can target anti-cancer bioactive peptides. We can target anti-inflammatory, any form of bioactive peptides. The good thing is that bioactive peptides spread across all possible forms. All that you can think about in medicine, we can get through bioactive peptides. So we, at this point, invite other interests who want to collaborate with us to move around. So now, when you look at this, you will see the tripod synergy. We have the indigenous fermentation, we have an AI-driven model, we have microfluidic. What does that give us? It gives us a context of good prediction, we have a reduced time, then we have the high throughput targets, all because we have a bioactive peptide that can be recovered within this. So if we join these three initiatives, we are going to have bioactive peptides that are recovered, discovered, characterized, and purified to give us a quick access to supplements and nutraceuticals. Now, the next thing is a regional innovation hub and policy integration here. We are thinking about how can we involve other parts of African fermented foods. Right now, we have been concentrating on Nigerian fermented foods, but Kenya, Uganda, Ethiopia, all other African parts have their specific fermented products. Sometimes the source of protein or source of material may be the same, but the fermentation protocol may be different. And so if we gather all these together and we are able to form this synergy, we are likely to have a very big innovative breakthrough. Now, the good thing here is that we are dealing with African materials for Africans and through Africans. So we can have capacity building, we can have commercialization, and these are the real things we are looking at. We want a situation where we can have researchers from any part of Africa coming together to look at the different forms of fermented products, and with that, have joint products cutting across Africa. If I can develop a product with somebody from Kenya, Uganda, Ethiopia, whatever, the product will definitely be in the market in Nigeria, in Ethiopia, and all that at the same time. So that makes it very easy. So we look at the ethical, socio-economic, and commercialization perspective. These are the ideas that I've spoken about. We need lots and lots of integration and collaboration. So here we have listed out some of the future of our expectations, particularly cross-disciplinary collaboration. We are open to, we have opened our arms and our laboratories for as many who want to join this great initiative. With this, I want to say thank you for listening.
Thank you very much. Let's give Professor another round of applause. And we can take 2 questions. We are actually short of time. So 2 questions. Thank you.
Thank you, Professor. My name is Patrick Mora, and I'm very interested with your project. And I've noted that your model is reporting very high metrics. But there is a slogan that goes that all models are wrong, but some are useful. So I'm concerned with the, for example, you have accuracy of 0.797. That means it falls short of 100 by 3%. So what might go wrong when we replicate this model? That 3% may become something, you know, the multiplication issue of that error. So what might go wrong whenever we have such kind of a discrepancy in accuracy? My second question was, now you're talking about food that communities across Africa ferment, and some of these foods are their cultural heritage. and culture sometimes, or maybe nowadays, is becoming protected. All right. So I'm wondering what might we experience with the cultural, mutually agreeable terms with use of your model. Thank you.
All right, thank you so much. That's a very interesting question. Now, first of all, I want us to understand that this concept is workable. And having started, it is open to further development. Right now, we are working with very limited number of fermented conditions and outputs. And of course, you agree with me that in machine learning, you need large data source. For now, that large data source is still in progress, and we're likely going to cross that when we optimize our various options and get the thorough puts, and then we retrain until we get the desired output, all right? As for the heritage, this is one area that has been passion for us. Africa has a lot of heritage, and we do not make noise about it until somebody across the ocean comes and discovers what you have been using. That discovery to me makes no sense. So we are using it, we are in it. So who discovered it? Do you understand my question? Who discovered it? And because we have been so quiet, it's our heritage. So all we need to do is to pick the traditional knowledge, which is your heritage, put a scientific refinement on it, use machine learning to grow it. We may need to modify what it is to get our output. At that point, it becomes a technology. It becomes an acceptable scientific technology. That's why we are calling for a research innovation hubs on this fermented product, AI and microfluidics across Africa, so that I may not be the only one doing it. You can be there. Another person can be there. All we need to do is to form a network which makes it to grow, and we still maintain our heritage. For instance, if I am working with a product from Ghana and we patent it as a Ghana product, it's still an African product. Our heritage is conserved. Unlike if somebody from across the ocean picks it and patents it, it becomes, you understand. And then I've seen situations where people have Nobel Prize for identifying what we have already been using. For me, it doesn't make sense. Thank you so much.
Thank you, Prof. I think we'll stop at that. Prof is around. You can follow up with questions. Let's appreciate him with another round of applause. Good. So I think we'll just proceed to the next talk, which I'm super happy to do. Good. Yeah, so I'm now not sharing, I'm now the presenter. So I'm super happy to
Oh, all right. So please, ladies and gentlemen, give me the pleasure to introduce the renowned researcher, an excellent teacher and educator. He has done so much in the point of care diagnostics. He's come up with novel techniques, and most of all, he's one of the most key supporters of this meeting. So Professor Jesse Gitaka is a friend of mine, but he works on quite a range of fields. On malaria, he has so many bases across the country, from Busia to Homa Bay. He has done so much on point-of-care diagnostics for infectious diseases, and he will share with us some of the greatest work he has done. So please, I don't want to talk so much about his so many records, but I will stop here and allow him to tell us what you have, the insights, please. Thank you.
Thank you, Ajoga. Yeah, so very flowery introduction, and yeah, I'm excited to be here. Thank you for the for this, especially for our KIPRE collaborators and co-conveners of this meeting. Jesse, as you've heard, and today I'll not be talking so much about our work, although I'll mention that briefly. I was talking about building multidisciplinary research capacity, basically talking about how we can work together to ensure that we build research capacity within our institutions. So I'll give the example of our institution at Mount Kenya University. And first of all, before I go any further, I really want to give a shout out to two of my colleagues, Lucy Mudhoni, who is maybe around here. And so she's a PhD student who actually made it to the top 40 under 40 women on the BD. So let's clap for her. And also our very own colleague, Dr. Bernard Kanoi, who is actually the current immunologist of the month. And as you can see down there in the description, he's the East African regional representative to the Federation of Immunological Societies. So let's clap for him. Yeah, basically, these two guys coming from our lab, I just want to show how the profile of what I'm just going to talk about, and you can be sure we're just behind there following on their footsteps. And so why do we need to think about capacity building? Okay, for research especially. So Africa constitutes 18% of the world's people. However, we produce less than 2% of research output. Okay, whether you're talking about patents or innovations or publications, in all those metrics, we are actually doing very badly. And we have a very low density, PhD density. In other words, if you are to count the number of PhDs in a country and then convert that to per capita, let's say per 1,000 people or per million, we are lagging behind. And that perhaps contributes to the big challenge that we see, where amongst other things, when it comes to vaccines, almost all the vaccines we consume are made outside of Africa. All the diagnostics we are using, all the drugs we are using, all the technologies, all the equipment are big. I mean, the figures are like below to 5%, below 10%. We are not equitably contributing to these products when you consider our population and capacity. So this is why we need to build capacity in Africa for research, so that we are able to contribute to our own well-being and to the rest of the world. Africa faces serious infectious diseases. We all know the burdens are huge: malaria, tuberculosis, HIV, neglected tropical diseases, and so on. Fast rising NCDs, non-communicable diseases. As you all know, this is a growing problem. And actually, if you look at all the metrics, we are doing badly when you consider to the other races. Climate-linked threats, this is something also for us to think about. We have a huge youth majority with limited high-skilled job pathways. Yet, as Tom mentioned this morning here, research and science and technology have the capacity to produce dignified, high-quality jobs. So capacity building enables local solutions, rapid outbreak response, innovation, and dignity in work. And therefore, it's super important for us to train and retain African scientists to drive health, science, and economic transformation. In other words, we have all the reasons on earth to build research capacity in our continent. And So in our group, these are some of the core skills that we develop. Of course, we do education and training as a university and also as a lab. We conduct basic research for discovery, genomic and immunological analysis, preclinical testing, especially in collaboration with KIPRE and others, clinical trials, design and execution, and we also do a lot of community engagement and policy development. Some of these things I'll be talking about briefly. So, okay, this thing is not moving. Okay, thank you. So one of the things I really wanted to talk about is the issue of placental malaria. So placental malaria is a uniquely African thing because as we all know that more than 90% of malaria cases occur in Africa. And this is a huge problem for us. You can see a huge percentage of pregnancies at risk of placental malaria. And this is despite having current interventions. We have approximately 10,000 maternal deaths due to this challenge of malaria in pregnancy, 200,000 stillbirths, and about 500,000 low birth weight babies per annum. So that actually tells you that from the time the baby is born, they are already disadvantaged. They are already coming to the world, minors, and the potential, of course, means a lot when you think about the socioeconomic status of the continent. And sorry, I think, good. Yeah, so there are things we know about placental malaria. Of course, we know about the burden. We know that placental malaria is majorly driven by oxidative stress and inflammation in the placenta that affects cellular and molecular disruption, including processes such as the inflammasome, autophagy, the innate immunity, impaired blood flow, and reduced oxygen nutrient transport. And all these, you can see, are more at the cellular and molecular disruption levels. We do not know a lot of things that will enable us to discover innovative strategies, including therapies, diagnostics, and so on. So we need to know how the host and parasite genes interact. We need to know how the host and parasite molecular pathways in placental malaria lead to these adverse outcomes that we actually see and I've talked about. And so we have opportunity to employ new strategies that were not there before. Spatial transcriptomics, single-cell RNA sequencing, and machine learning, as you already people have talked about. In other words, we need to leverage new technologies to understanding this problem properly. And so my group is working on this. We just secured some funding to help us tackle that problem. And one of the key problems we want to address out of all this quagmire is placental malaria diagnosis. So you can imagine a woman in Bungoma or in Kisumu or Homa Bay. She's pregnant 6 months or 5 months. And one day she just realizes the baby is not kicking anymore. She goes to the hospital, tells the nurse, I can't feel the baby kicking. And soon they listen to the fetal heart, it's not there. It's a stillbirth. A few hours later or the following day, the baby is expelled. But it leaves the questions to the mother. What happened? What did I do wrong? But actually, if they look at the placenta, they see the parasites. So it means placental malaria, which actually the mother will not have the symptoms for. The mother will be okay, going to the market, doing anything that she's doing. She doesn't have a fever. She'll not feel weak. It's just the problem affects the fetus, directly, and as you can see. So, and this is not the only issue. I talked about stillbirth. We're talking about low birth weight deliveries, IUGR, intrauterine growth restriction, and so on. So, how do we enable testing at the point of care in the facilities so that mothers can be put on treatment promptly. So if you look at the gold standard diagnosis for malaria, we're basically talking about blood smear microscopy and RDT, which is routinely done, especially, they can see parasites in the peripheral circulation. However, these gold standard strategies, they need highly skilled personnel, subjective, labor and cost intensive, and they miss asymptomatic cases which cannot detect placenta sequestered infected erythrocytes. And something interesting I need to mention, these placental malaria infected parasites actually have unique proteins that enable them to sequester, to stick in the placenta. So for placental malaria, the gold standard is actually to do placental histology. But placental histology means you need a piece of the placenta. So how can you acquire the placenta during gestation, during pregnancy? It's a risk-invasive procedure, which is actually not possible. Okay, so we therefore need better tests that will pick sensitively the infection in maternal blood, whether you're looking at pieces of the parasite or products of that infection, that pathology. This could be like inflammatory signatures and so on. And so we have a rich standardized biobank resource to enable us to do this. This work actually led by my colleague, Dr. Kobia, who is seated at the back there. Cutting edge samples, remember I'm talking about capacity building. So one of the things to build that capacity is to really have resources that researchers can leverage more cost efficiently, because once we collect these samples, put them on the repository, other people can leverage on those. So we are able to do RNA studies, protein studies, histology, and others, and make correlations or even discovery using these samples. And so this is just a demonstration of how the placental malaria is detrimental to the baby. As you can see, there's significant difference in the birth weights of babies from mothers who did not have placental malaria and those who had. Okay, that tells you this impact is big. Even the placental weight itself is affected. Okay, and yeah, and this is one of my favorite slides, just to show you how complicated and bizarre this problem is. If you look at the outcomes, okay, so here you have 179 placentas. You can see that the level of parasitemia varies hugely, from something 0.1 or 0.2% all the way to around 35%. Okay, there's huge variability, and interestingly, women who have high parasitemia does not mean necessarily they'll have adverse outcomes. They may have low parasitemia with adverse outcomes or high parasitemia with normal outcomes. So that shows there's complex underlying mechanisms that we need to decipher. So this is a key thing for us, and I'm super happy to say that now with the resources in our lab, we are able to tackle some of these problems. Okay. What does this thing first? Okay, sorry. Good. So the other things my group is interested in is diagnostics development for other infections, especially those that still affect mothers. We are looking at placental glycan as a biomarker for placental malaria. We've done a COVID-19 lateral flow assay test. We are working on mpox assay, an assay for TB, Neisseria gonorrhea, chlamydia trachomatis, Trichomonas vaginalis, and syphilis. And actually, we have many of my colleagues from the group who will be presenting posters. Please watch out for this. as well as talks. I know some of them were done yesterday. So please find opportunity to have a discussion with them, and we are super happy to collaborate. We also do work on malaria surveillance for, evidence of resistance. We know countries around Kenya, including Uganda, Rwanda, Tanzania, have detected this artemisinin resistance. And so we also found parasites in Busia that have this C469Y mutation, which actually helped us to really inform the National Malaria Control Program about this challenge. And actually, I'm happy to say my colleagues in Kilifi as well as in Kisumu have taken up this work, and they have actually, brought more evidence to show that we are facing a serious threat as a country. So this is evidence of contribution to policy and how we can help our Ministry of Health to best be prepared for this kind of eventuality. Okay, so the other thing that we are very passionate about is malaria vaccinology. I know Dr. Kim talked about this yesterday. So this work is actually led by Bernard, who you'll be seeing many times around here. And he's employing immunoscreening and reverse vaccinology strategies together with domiciled wheat germ cell-free system that we now have in our group. So you can, if you need, well-folded, well-designed proteins coming from this system, which is actually superior to E. coli or even other systems, you can engage with us, and we are happy to collaborate. And so this is part of that work again, and I know this manuscript is actually published already. So just showing target discovery for vaccines, for malaria vaccines, and this is actually super interesting work as well. So if you're keen, please link with us. We are happy to share more. Other programs in addressing pregnancy complications. So we are soon rolling out a clinical trial funded by the Gates Foundation to try nutritional supplements, L-arginine and L-citrulline, for prevention of preeclampsia, in a way, trying to combat the adverse outcomes of this challenge. We are also keen on group B streptococcus, which is also a major cause of neonatal sepsis. And neonatal sepsis, we know, leaves adverse sequelae on the newborn. Okay, so and for us to do so, we need to understand the bacteria, develop diagnostics for it, and soon there are going to be vaccines for it as well. So we have a thematic area as a focus for group B streptococcus as well. And we're also leveraging innovative approaches in building clinical decision support for both newborns through this IFIA neonatal care application, so which leverages on language models. So you can, if you check at ifia.co.ke, you can download the application, and this actually has finished the pilot testing, and we are keen to proceed to phase 2. We've also done a clinical decision support tool for non-communicable diseases under that address, and we know some people may be interested in this, and we are super happy to collaborate on that as well. Research infrastructure, you cannot have capacity without infrastructure. Infrastructure is fundamental. So we have, like in this photo, you can see our team is all smiles following the installation of the NextSec, the Illumina NextSec sequencer, I think approximately a year and a few months ago. And so those are some of the things I think I've already mentioned that we are doing in the unit. And from here, I think I'll just make the presentation even lighter, showing you some of our, both local and international human capacity engagements. On the last slide there, you can see Ronnie with, engaging with patients in Bungoma, We have our nurses in Thika. Here is just some of the people who chose to political careers or administrative careers enjoying time in the lab. And there, the group of students and collaborators who had visited, engaging in research. There's Clement and the team working on something. This is a poster for a parasitology workshop we had. And here we had a visitor, Faith. She presented yesterday. I had visited our group, and we just taken her around the lab tour. In other words, our research infrastructure capacity is at the top level, cutting edge. Kennedy can confirm this. And we are super happy to engage with everybody here and to sort of do more and achieve more. So this a group photo with some visitors from Japan, from the United States, and we also do, I think I've mentioned this, capacity building through policy engagement. This is Dr. Kibor and his team, Dr. Onyango, from the National Malaria Control Program. So we do a lot with them. We have we've undertaken studies on their behalf. Our studies have informed their work, and here they just visited the lab, and there we had visited them in their office to engage more. And it's not all lab. Sometimes we get out of the lab to unwind. So that's some members of the team. I think they were just preparing to do some athletics or football. Mary can tell us what they were just about to do. And here, as you can see, me and some other guys were just jumping up the Mfangano Island mountains. So, in other words, I'm trying to say we have, through collaboration with our partners abroad, locally, developed some research infrastructure, research capacity that would like all of us to tap into as we address the myriad of challenges that we face as a continent. We acknowledge our collaborators and funders. Thank you very much.
Wow, Ayoga, that was really powered, and thank you so much. Great, so when you talk about cutting edge research, and when you talk about research infrastructure, I must confess that Professor Gitaka leads, and he has one of the best labs I've ever visited across the continent. And I must say that congratulations for doing the great work that you're doing. So I will open his talk for questions. And anyone with a question for Professor Gitaka, please, we can pass the microphone so that you can tell us your question. Anyone? Okay, as they think through, perhaps I want to get back to the subject that you touched on, the capacity building. And I must say that we had a fantastic time with you at MKU hosting the workshops here. And thank you so much for that. So it's something which is very, very critical to building that pool, the critical base that will support the R&D. And I think we heard about the vaccine development and quite a wide range of tools and innovations that we need to support the challenges that we're facing in the continent. So I just want to hear more about your perspective. What are the challenges that you see, and how do you think we should be able to address those in terms of capacity building and in the continent?
Okay, thank you. Yeah, so indeed, there's nothing that comes without its own challenges, and but the challenges are there to be tackled. So foremost, I think we need the right mentality, the right attitude. We need great ideas. Of course, people may want to talk about funding and the challenge of funding. We all know the ecosystem has changed. But my take is that despite this being a challenge, it is actually surmountable. You can overcome it. And as Tom mentioned in the morning, there is, and you could see from the resources they have, there's a lot of resources actually available, okay? It's just that these resources are going to a few groups within the continent, mostly, going down to South Africa or to Egypt or to, a few places in Kenya and so on. So I think people need to focus on cutting edge, great ideas that whenever a funder looks at, they just, open the pass for you. So I think our mentality, attitude, is more important actually. The money is not really the big problem. The money follows after we sort the ideas and this kind of thing. The other thing is, and this I've seen with colleagues of mine, we need institutional support. Some institutions, they all talk about research, capacity building, but they actually do not have the skin in the game, as you say. They do not put the resources to facilitate that. This could be through weak systems in the universities, and so this is a problem to be addressed. It's a serious one actually, where our universities and research institutes are actually tinkering on the blink of insolvency. When a university is not able to pay its salary, salaries for lecturers, you can imagine it will not have resources to put into research. This is the reality of the situation. So this is another thing to be sorted out. I think we need more funding, certainly, from the government to support those institutions. And then those institutions also need to have the right people. We need more young people interested in research. We need advocacy. We need to talk to our MPs and whoever, senators, so that we have more money put to research. Otherwise, I think all these problems, when we work together, are things that are surmountable. Thank you.
All right. Thank you. So when you talk about mindset, is a very critical thing, and I just want to pass it over to the professor, please.
Thank you very much, Prof, for that presentation. I want to address the issue of sustainability. I find sometimes we have these kind of equipment, but they go down with the end of the project. Like if it's a sequencing capacity, you run the samples in your project, but after that, that particular equipment more or less is for training, but not for research. So that aspect of sustainability, maybe you can address it. And you find that now every other institution wants to buy a sequencer, and eventually these sequencers, are treated the same. You don't have enough samples to run the equipment, such kind of a thing. I don't know how, as a country, generally we can address that issue. Yeah, how we can harness our efforts together. I don't know, maybe you have some thoughts. I don't know.
Very great. I know the question is for you, but I will share my experience outside there. The thing we do, they stopped funding individual labs to buy equipment. The thing is, the university must set up a shared common facility, and everyone who applies for funding, you must make sure that the equipment you buy, it goes to that shared facility, so the whole institution has access to it. You share the consumables, but the equipment belongs to everyone in the institution. And sometimes you open it up for all the country and sometimes the region. So anyone who has, if you have a flow cytometer, somebody from Nairobi University, from JKUAT, will have access to it. You charge them the consumables. So that's how it's run in other parts of the world. But I want to hear from you, please.
Yeah, I think you just read my mind. That's the answer, actually. For us, our resources are open. Our lab is open. Our sequencer, actually, I actually tasked the supplier who brought it. I told him, now go out and preach the word, and we are open. We know this is a problem, especially for some of the institutions who close the door for other people. For us, we are open. We do not have this colonialist mentality. of overcharging local people who want to use the machine or to do sequencing in our lab. So we are game, we are open, and we are inviting everybody to come use our facility. And actually that's what is happening, but we would like to see more of that. So I agree, sometimes sustainability actually is a big problem, and for my team, I actually tell them, for me the biggest headache is how do we remain sustainable? How do we ensure that we are able to run beyond the grants? So, so far I think with this kind of thinking, we are seeing a change, and actually we are seeing research organizations warming up with the idea and coming to work with us. We also do not segregate about students in my lab. We have students from KU, Kenyatta University, University of Nairobi, JKUAT, wherever they come, Maseno University. We do not just take students from Mount Kenya University. We are open. We are even open to students from abroad. Okay, so we have had students from Ghana, from Tanzania, from Burundi, and so on. So the idea is to, as you bring in more people, as you have more institutions coming in, you even become more attractive in terms of, getting the money to run the facility. Thank you.
Great. T is waiting for us, but if anyone has another pressing question, I think we're going to take the last one. Okay. I know, Prof, here, this is passion.
Yeah, again, from experience, and I know all of us have had some experience when it comes to surprise. How are you managing to get your surprise in good time? And do you have companies of choice? For example, if you want molecular biology reagents in 2 weeks in Kenya, in the environment that we are in, is it possible?
Yeah, so nice question. So actually, the secret is proper planning. This is the secret, and having a relationship with the suppliers. For most of the molecular reagents, actually, we know that there's a lot of it's called single procurement or something. You cannot just ask anybody to bring you an enzyme. Most of those enzymes will also will be brought in by, let's say, ABL or FNS, or depending, Change Biotech or whoever, depending on what rights they have together with the main supplier. For instance, if you for the sequencing equipment, the Illumina, the company we work with is FNS, because they are the sole distributors of that company. So our strategy is to work with them and to anticipate our supply needs, to order in good time, and to sometimes always keep a buffer in the freezer. So this is how we've gone. We are coping with that. Sometimes the biggest challenge actually is not the lead time between you and the supplier. It is between you and the LPO. The institutional procurement people. So we've been able to overcome that challenge, majorly because we are a private university. We are not under the shackles of public procurement. It's called people. What is it called? Public Now there's e-government. We are not under those kind of. We are not under those kind of challenges. When we want something, we just get a requisition, is signed by myself or my colleague, Dr. Bernard, and our procurement manager, lab manager Mary, orders for it. The company brings it, goes through the store, they stamp, it's landed, then we write for them a check. I mean, we a requisition so that they can, an impress so that they can be paid. And this way, if a company delivers today, let's say on the 15th of the month, by the end of the month, they have their money. So these companies are actually now becoming, they love us more than anybody else, because they are able to get paid in good time, and the work is able to proceed without delays. Thank you.
Great. Thank you very much. So I think with that, we shall end the session. But before we end, I just want to first, once more, please, let's appreciate Professor Gitaka. Thank you so much.
Oh, Ken tells me it was my session, but he has already done the needful. So we have a 20 minutes break, right? Thank you. Let's get moving.
Thanks. This one. Excuse me, the young investigator sessions, if you're here, please, we need you to upload your PowerPoint immediately. Young investigators, I will call your name, Protas, Charmaine, Samuel, Grace, Charles, Winnie. They are JC's students. And once again, young investigator sessions, please speakers, upload your files. Thank you.
let's settle down, please, so that you can begin. So this session is for young investigators. The second part, the first part was done yesterday. So we'll have the 6 of them. We'll have Protas. Where is Protas? Kindly, let's sit there, in that table over there. Shamim. Where is Shamim? Let's sit there. Samuel Nganga. Where is Samuel Nganga? Is Samuel Nganga here? Okay. Grace Okicha. Grace Okicha. I saw her. Grace Okicha. Charles Kivasu. Charles Kivasu. Please, let's all sit in that table there. And please make sure that you've uploaded your presentation with the media team. Winnie Kulei. Where is Winnie? Okay. As Grace come in. Grace Okicha. Okay, please occupy that table. So we'd like to begin. So these are the ground rules for these sessions. So we'll have 7 minutes of presentation and 3 minutes of questions. So the first, we'll do the first 3 presenters. That is Protas. We'll start with Protas, followed by Shamim, followed by Samuel. Samuel, come in. Samuel. So that will be the first 3. Then we take questions. Then we'll follow with this Grace, Charles, then Winnie. So remember, it's 7 minutes. So we'll remind you when you have 5 minutes that you have 2 minutes to go, so that we have time for questions at the end. So this is how you'll be evaluated on time management, how you present, and how you respond to the questions that are asked. Okay? So can we begin with the Protas? Please take the stage. Before we begin, sorry, I didn't mention my name. My name is Martin Omondi from Kenya Institute of Primate Research, and I will co-chair with
Dr. Rachel Kimani from Mount Kenya University.
Good evening, everyone. I'm Protas Omondi. I'm from the Center of Malaria Elimination, Institute of Tropical Medicine from Mount Kenya University. And yesterday we saw some good talks on malaria, and most of them, they were emphasizing on some of the stage-specific stages that can be leveraged to come up with interventions. So for my study, I will talk about one of the most important stages of the Plasmodium falciparum, which is the gametocytes. They're also known as the sexual stages of the parasites. So this project was under SATREPS, which was for malaria-free in Kenya. It was done in the Lake Victoria region in Homa Bay. So this is just a small part of the study that I did during my PhD. So in the introduction, I will emphasize that, as we know, the the life cycle of Plasmodium falciparum is complex, and it alternates between the human host and the mosquitoes. And one of the important stages, which I've emphasized here, is the gametocytes. And these gametocytes are very important because they are the ones that enable malaria transmission or transmission to mosquitoes. So for a successful fertilization, we must have both male and female gametocytes. So I must emphasize that these are the sexual stages which they originate from the asexual stages. So gametocyte identification is it's very challenging because we cannot rely on microscopy. So these are usually detected through the stage-specific transcripts, which are only expressed during these stages. As you can see, gametocyte has 5 stages, and only the mature stages can be seen on the peripheral blood. So we usually use RNA-based methods to be able to detect these gametocytes. And it is important to note that for an individual to be to be transmissible, they must have both the males and the female gametocytes. So moving forward, so when I joined this project, the SATREPS, as a PhD student, one of the major problems that was encountered in the region, it was that this region has a very high and heterogeneous prevalence of asymptomatic malaria. And most of these infections are also submicroscopic. So given that I had some experience with these gametocytes, I was interested to see how do these gametocytes play a role in this community. And most importantly, the school children who are the most vulnerable, and also they are the key transmitters of malaria in the region. So we decided to evaluate these gametocytes using the methods which I will explain. Sorry. Please note of that. Sorry. Okay. Okay, this is good. Thank you. So we did a cross-sectional school survey in the entire Lake region from schools. And these were about 27 schools. And we were able to do malaria detection overall. And for my case, I was majorly interested in the gametocytes. So I developed this design so that I can be able to come up with a way where I will collect whole blood. Because I've mentioned for gametocytes, we have to target the RNA. So moving forward, we have done quite a number of cross-sectional surveys in the region. And mainly we collect whole blood samples, dried blood spots, and of course blood smear for microscopy. And then so that we are able to determine the overall prevalence and also to compare how the submicroscopic and microscopic are in the area. So this is just the analysis that I did, and we did a couple of regression models to be able to identify some of the risk factors that affect the dynamics of these gametocytes and also the sex ratio. So going to the results, I will just mention that in the characteristic of the study population, we were able to recruit a total of 1,639 for this specific study. The median age was 8 years. As you know, it was school children. And malaria-related fever was just 4%, meaning majority of them are asymptomatic. And only 54% reported to have slept in under the net the previous night. This is kind of surprising because we expected at least majority of the children to be sleeping under the net. So for the results, we can see that the prevalence by microscopy was 7.2, by PCR was 21.7, which is expected to be higher compared to microscopy. And one important, most important thing to say is that majority of these infections were submicroscopic and asymptomatic. You can see 80% of them were asymptomatic, and 66% of these infections, those who were positive, were submicroscopic. So now, looking at these positive infections, nearly half of them, they carried gametocytes. This is a serious problem because this translates to transmission potential. Of course, there is a way to confirm how infectious they are, but this shows how these kids, how these hidden infections carry gametocytes. And higher gametocytes was also found in children under the age of, over 5. So under age of 5 were mostly not carrying gametocytes. As we know, they are always protected by, because they're still young. Also, some of them have received the vaccine, and most of them, their parents are usually keen to ensure they sleep under the net. And looking into the gametocyte, so usually, the gametocytes are usually, we have both male and female. But for a successful fertilization, one male differentiates into 8 gametes, and it's able to fertilize 8 males. So it was female biased as expected. However, this sex ratio was influenced by several factors, which I will talk about. So what this means is that transmission advantage is well noted, and this female biasness confirms that indeed these individuals can result to transmission. And looking at some of the host and parasite factors that drive this variation, school-age children under over the age of 5 and those with PF monoinfections were the major reservoir of these gametocytes. Anemia was associated with higher gametocyte densities. And if you look at the proportion of male gametocytes, we found that the proportion of male gametocytes, this was very crucial.
Please wind up, time is up.
Yes, it was decreasing compared to, it was low when in low densities, the proportion of gametocytes were higher. This means that in lower densities where the transmission risk is reduced, these gametocytes are able to produce more males so that they can be able to fertilize all the females. And in conclusion, most infections were submicroscopic and asymptomatic. Gametocyte density was affected by host and parasite factors as well as sex ratios. And in the next step, we want to link these proportions of male gametocytes into mosquito infection through a membrane feeding assay. And in recommendations, we recommend these school-based surveys should be done regularly, expand diagnostic tools to be able to capture these infections, target gametocytes using various strategies that have already been mentioned, and most important, to strengthen community surveillance. And a practical example is this region called, is this island Mfangano. We can see the prevalence of gametocytes was very high in the region, and this indicates that if we are able to target this region, for instance, with treatment and vector control intervention, we can be able to eliminate the malaria. So thank you. I would like to acknowledge all these people and the institutions that helped me to finalize the study. Thank you very much.
Thank you, Protas. Our next presenter is Shamim Wanjiku.
Good evening, everyone. I'm going to present on sickle cell anemia and severe malaria anemia in children under 5, particularly findings that we have obtained from some of our studies. So malaria is still endemic in 83 countries. And in 2023, there were 597,000 deaths. And of these deaths, 76% were attributable to children under 5, particularly in the WHO Africa region. And so our region of interest is Western Kenya. And in this area, malaria has the highest prevalence in the country. In this area also, severe malaria manifests as severe malaria anemia in these children under 5. I'll also so I'm going to also talk about sickle cell anemia, which is caused by a mutation in the hemoglobin beta gene and results in the formation of defective hemoglobin, which is which are sickle shaped. And this leads to impeded blood flow in the vessels. And some of the symptoms of this disease are pain crisis, chronic hemolytic anemia, and complications such as stroke and chest issues. So globally, 7.7 million estimated people live with sickle cell anemia. And of the 515 new births, 80% are in sub-Saharan Africa. So sickle cell anemia is quite a huge burden in this area of Western Kenya, sorry. You can see in this map, the areas that have malaria also have sickle cell anemia. And this is because the heterozygous state of the gene results in protection against severe malaria. So our objective was to characterize the entire expressed blood transcriptome in children with sickle cell anemia and severe malaria anemia to see if we can identify molecular signatures and then find therapeutic targets, potential therapeutic targets. So for that, our study area was Siaya County Referral Hospital in Western Kenya, as indicated in the map. And we also got approval of ethics from Maseno and the University of New Mexico. So we enrolled children between 2017 to 2020. They were categorized into two. So there are those who had severe malaria anemia and those who had non-severe malaria anemia. And then my focus was on the ones with severe malaria anemia, which was stratified into sickle cell anemia and those without sickle cell anemia. So we had 7 with sickle cell and 18 with non-sickle cell. And on those, we performed gene expression analysis and pathway enrichment analysis. So this table shows the demographic and clinical characteristics of the participants. Just to highlight that the underlying parameter, the underlying hematological parameters show the underlying pathophysiology of severe malaria anemia, which is characterized by chronic hemolysis, bone marrow compensation, as you can see, the red blood cells are lower in sickle cell anemia compared to non-sickle cell anemia, and also inflammation, you can see with the white blood cells and the lymphocytes. These are the significant hematological parameters. We went ahead and did gene expression analysis, which was we had over 53,000 transcripts. And of these transcripts from the volcano plot, the red ones are the upregulated. There were 2,110, and the blue ones are the downregulated genes. In these children with sickle cell anemia, severe malaria anemia, which contain those with the sickle cell anemia and those without the sickle cell anemia. So from the upregulated and downregulated genes, we did unsupervised classification, and we had 3 clusters that emerged in these. So these were the top 1,000 differentially expressed genes, and we had 3 clusters. So from these clusters, we can see the first one was downregulated, the second was upregulated, and the third was slightly much of a mix. And from that cluster, so we wanted to understand what these clusters contain, and we obtained this is the first network we obtained from the first cluster, which contained downregulated genes. The blue dots, the blue circles show the downregulated genes. This network was obtained using a tool called Metaco, and we have the box. The black box shows the divergence hub, and the red box shows the convergence hub. So CYMYC is the transcription factor in charge of stress control, stress and hypoxia control, and FC alpha is also a transcription factor, but in the immune system. And this network reflected that the immune system was downregulated, and it was suppressed in these children. And then just to understand what the network, because the network is robust, we wanted to understand what terms would emerge if we were looking for the pathways. And we did a gene ontology, and we saw that this diagram shows some of the terms that emerged when we did gene ontology. So it reflects the immune system was downregulated. It was suppressed in children with sickle cell anemia. We also did, for the second cluster, which contained upregulated genes. I know this is hard to see or read, but there was this red and black box which contains ubiquitin. Ubiquitin was the convergin and the divergence hub, and ubiquitin is a core protein in the ubiquitin proteasome system, which is very key in the degradation of proteins, as well as in controlling stress response in cells. So this was upregulated in the children with sickle cell anemia. We also did a gene ontology on the same cluster, and it also reflected terms that were associated with the ubiquitin proteasome system. We have mitophagy, ubiquitin binding, ubiquitin-like protein, and this shows that these children were, they had increased stress response. In conclusion, we had the 2,110 upregulated genes, 710 downregulated genes in children with sickle cell anemia and severe malaria anemia. And then we also, the downregulated genes showed this network that was linked to reduced immune system activity. And the upregulated genes showed this network that is linked to upregulated degradation of proteins and stress response. So, yeah. And the GO pathway analysis also reflected the same. I'd like to acknowledge the University of New Mexico team, Kenyan Global Health team, all the children who participated in the study and the guardians, and the NIH for providing funding for this study. Thank you.
Thank you, Shamim. I'd like to invite Samuel Nganga to make his presentation.
Good evening. I am Nganga Samuel from the Center for Malaria Elimination, the Institute of Tropical Medicine at Mount Kenya University. And we'll be looking at, in the next 7 minutes, at temporal patterns of PF exposure, where we are looking at antigen-specific antibody kinetics and also effect of rain. The speakers before me have given the figures. Indeed, malaria remains a global health challenge. Malaria is still endemic in Kenya, as you see from that image. So we have efforts globally. We have chemotherapeutics, we have insecticides, we have respiratory vaccines. However, efforts towards malaria-free world remains a challenge, and therefore that's where serology comes in. But even as you look at serology, naturally acquired immunity to malaria develops over the years of repeated exposure. This process is not fully understood, it is complex, and so we have to target multiple antigens. And this is because in malaria, especially PF malaria, we have over 5,000 antigens. Therefore, there is need for novel surveillance tools to capture dynamic host, parasite, and environment interactions, as Professor Jesse Aria talked about. We therefore included multi-antigens, that is pre-erythrocytic and erythrocytic antigens, And these are the questions we wanted to answer. How do antibody responses to different antigens accumulate with age? Are there sex-based differences that influence the development of this immunity? And which antigens can we use as indicators of recent versus long-term protection? And also include the aspect of environment where we looked at rainfall. And so those were our three objectives. Quantify the temporal infection dynamics. We also had PCL data, and therefore look at PCL, whether it correlates with antigen-specific antibody kinetics for pre-erythrocytic and blood stage targets. Secondary, look at heterogeneous antibody persistence, and also identify, do we have antigens that cluster, that is pre and post, or rather erythrocytic life cycles. And lastly, to determine antigen-specific environmental driver, and that this is rainfall. This cohort was earlier introduced yesterday in the morning by Dr. Mtaka Ingara. In this pilot, we only took 162 individuals. It is a cohort of about 500 individuals. These serum samples are at the Mount Kenya Biobank, and we had serum samples from these 162 individuals over 13 sampling points, that is in an year. My cohort is from Homa Bay, Western Kenya, a malaria endemic region. We used Luminex for the study design, Luminex multiplex assay design, which is also flow cytometry based. So Luminex 200, the image down here, is a multiplex, has the capability to do serological assays where we multiplex the antigens. So in our case, we had sporozoite, liver stage, and merozoite, 6 antigens. This system is in our lab. You are able to multiplex up to 100 analytes in 1 microliter of blood. And from the image above, you see we have a red razor to identify the antigen, the green to identify and also quantify whether we have the antibodies. How do we do coupling? Because we have the magnetic bead and you also have your protein. These proteins, as Dr. Kanoi earlier talked about, we have the wheat germ cell-free system established at MKU. You make your proteins, and then in the morning, you do in-house protein coupling. The magnetic beads have a carboxyl end, antigens has the amine end, and you're able to have a carboimide bond. So for the results, our cohort, we had individuals who had multiple infections and those that had 0 infections, or other infected and non-infected. What do we see from this graph from all the antigens? That those with repeated infections had a boost, and therefore they sustained higher antibody levels. The decay, as you observe, especially for MSP and MCP, the decay was slower, showing that there was immune reinforcement. Obviously, over the 13th sampling points, we had repetitive exposure. When we looked at antibody responses via age, so the first thing we did, because our cohort had samples that ranged from below 5 to over 45. So we had the 5 and belows, my young children, the school-going children between 5 and 15, and then the above, I classified them as adults. So the first thing we see, there is a rise, cumulative exposure to malaria antigen, which indicates age-related antibody, related immunity. The CSP and MSP had the lowest seropositivity across all age groups. And then we had CELTOS and LISP, which are pre-erythrocytic antigens, where we had the seropositivity intermediate and highest in MSP1 and also AMA1. So in this second image, it indicates, and I also have the p-values, that indeed the relationship was significant. So there is actually clustering, MSP1 and AMA, and the pre-erythrocytic antigens also cluster. So this looks at the breadth, and this image figure excites me because it shows that from the differences, a H index of 69, quite high, and also the p-values was below 0.05, which shows that immunity breadth increases with age. If you look at the very young, before even they are 5, they all already have a huge number of antigens which indeed are there. Therefore, there is rapid acquisition of initial immunity. And so we have to ask ourselves, given that we have the enrollment or rather deployment of RTSS, what is its impact? Because as we vaccinate these children, they also have very high baseline anti-CSP immunity. Now this are just a correlation matrix. Maybe the important thing from the first image, the overall, is that we had positive correlation between PCR and all the antigens, quite high. However, it's only MCP that indicates that is an indicator because it had a p-value which was significant. That's an indicator of recent exposure. From these other three, after I grouped the data, maybe the important thing is that for the young children, if you look at the first column for PCR against the antigens, we have positive correlation indicating that indeed it is the recent exposure, the infections that are driving this immunity, obviously because they are naive. If you look at the adults, the first column, negative correlation, indicating it's not the infections that are driving this immunity. Obviously, it is the accumulative antibody levels. So in this second last slide, I show the differential seasonal antibody kinetics. So I classified the rainy season, and the second, the month that preceded, succeeded, is the 2 to 4 week lag. That's where the antibodies rise. And if you look at that graph, they all had positive correlation showing that indeed there was a boost after the rainy season. During the rainy season, we have a lot of mosquitoes, and therefore, but most importantly is that for CSP, yes, it had a positive correlation showing it's an indicator of exposure. However, it was not significant. But for MCP, or rather CELTOS and RISP, which are pre-adipocytic antigens, they show stronger seasonal fluctuations, suggesting they are better indicators of recent exposure. Lastly, I looked at the sex differences, one of the objectives in malaria immunity, where we have either blue or red. That's where we have significant significance. And for blue, it indicates that the males have higher antibodies than the females. Red is the opposite. Now of importance, maybe what you're supposed to capture is a CSP, the first column, that in all, in young children and the small children, the males had higher antibody levels, but these reverse in adults. And I have given this citation. We had mice that were immunized with irradiated sporozoites, and in the adult females, there was a boost in anti-CSP antibodies. This indicates, this aligns with known sex hormone effects that during puberty, because of the estrogens, there is a boost in the anti-CSP antibodies. The other thing is that the sex differences in...
Time is up.
Okay, maybe the last thing before I want to acknowledge the study volunteers, the collaborators, Homa Bay County, where we got the samples, and all these funders, and my host, Mount Kenya University. Thank you.
Thank you, Samuel, for that presentation. We are going to have the Q&A session for the three. That is for Protas, Shamim, and Samuel. Anyone with a question?
Hi, I have a question for Protus. Why can you only detect the RNA in the gametocytes?
Thank you very much. So, like I mentioned, so in order to be able to differentiate between the asexual and the sexual stages, the gametocytes, they usually have, they express a lot of messenger RNA proteins, which are basically important for their development and differentiation. So we target those specifically, those specific messenger RNA as the marker for specific sex. So the male and the female. Thank you.
Thank you, Protas. From your presentation, you showed that children below 5 years were not majorly affected, and that is why you excluded them from your work. And the person who followed you in presentation was very keen on the children below 5 years. Now, from our paper, I realized that it was of a significant figure. And a lot of research is going on for the children below 5 years. Now, how true is this statement?
Actually, that is not true. I did not exclude the children under the age of 5. What I said is that those children under the age of 5, the odds of carrying these gametocytes was low. So this is very, it's true because you see, when you're talking about the odds of carrying gametocytes, we are trying to say the chances that this child is infected with gametocytes. As we know, school-age children, especially those who are like over 5, mostly they sleep, they don't sleep under the net, they sleep on the couch, so they're usually exposed to mosquitoes many times. But the under 5, what usually affects them mostly is the parasite density because of immunity. So for the case of this, the gametocytes were lower to the children under the age of 5. That is what the research says. Thank you.
Thank you, Protas. Any more questions?
Thank you. Mine is to Shamim. The transcription profiling that you did, the signatures that you're seeing, are there some interventions that are already existing maybe in other fields or any interventions that you would think about that can be exploited based on the results that you saw for the sickle cell?
If I got your question right, is you were asking about interventions for sickle cell from what I've observed or existing interventions?
Yeah, basically, how does your data inform our future management strategies?
So this is still ongoing work. We have not yet pinpointed a particular target or identified molecules to target that particular signature. But we are hoping to get to that point. So we are not yet there.
Thank you. Other question? Any question to Samuel? Okay, thank you very much. So now we want to go to the second presenters. So the first one. Okay, let's appreciate the 3 presenters even as we welcome the next. So the next presenter is Grace Okicha. So just remember 7 minutes presentation, 3 minutes question time. Then we'll remind you when you have 5 minutes through, and then again, time is up. Okay.
Good evening, everyone. My name is Grace Ndekiti. I'm going to present my master's thesis on examination of innate lymphocyte and innate like T cells in multiple sclerosis. This project was conducted at the University of Perth, Department of Immunology and Biotechnology, and supervision of Professor Peter Engelmann. So, multiple sclerosis is a chronic autoimmune disease of central nervous system, and this include brain and spinal cord. Usually, the etiology of this disease, exact causes is not really known, but we have several risk factors that are associated. Age and sex being the main one, in which the female population tend to affected more than male. And also we have genetic factors, environmental factors, and also some others like smoking and obesity. In multiple sclerosis, immune cells attack myelin sheath, which is a protective cover of the nerve fibers. This will lead to a breakdown of communication between brain and the rest of the body. In the case of uncontrolled attack, eventually patient will have a sensory or motor, and in a chronic condition, we can have cognitive. Globally, over 2 million people are estimated to have MS, and with the region away from equator tend to have higher prevalence, part of North America, Europe, Australia, and to narrow down to our immune cells of interest in this project, we worked with innate lymphocyte and innate like T cells. And each of these cells are classified based on their specific unique markers. And as you can see from their slide, I don't need to go through all of them. And my aim for this project is we want to study like comparison of innate lymphocyte and innate like T cells in multiple sclerosis and healthy control. And also further to investigate the changes in the population, total population, and the subset cells in each of the cells that I've mentioned. To do this, blood samples for this project was provided from the Department of Neurology, University of Perth still, and we were able to do the a standard FICO procedure to be able to isolate our PBMC. And cell counting is done, and then from the cells that we were able to get, 1.5 million we analyzed for their flow cytometry. This we did by each cell received a mixture of 9 antibodies which are conjugated with fluoroforms. And then we did the flow cytometry analysis. And also, because the project I've carried between June and June 2024 and February 2025, we had to sample several sample for the analysis, and some of the sample we stored, we froze them, and in case we do not have fresh allocate, we can always use the frozen sample to do the analysis. For the analysis part, we did flow cytometry, as mentioned, and then for the flow cytometry, we had to identify our cells of interest. To do that, a gating strategy is done. And for our cells, from CD45, we were able to get NK and NKT cells, and further, these cells are characterized or expressed in terms of CD4 and CD8. And my result, the NK population shows higher variability in the MS group for the total population and than healthy control. And when we go to the subset, we can observe there are CD8 positive in both groups, levels are elevated. Why do we say this? So we can explain based on the cytotoxicity and inflammatory response. So the cells tend to be activated because of the inflammatory response, and NK cells are elevated. We can also say based on the stage of this disease. In the chronic cases, we can have elevation in the NK cells. NKT cells, we also see variability within MS group and then LT control, and also for the subset group. For iNKT, myeloid cells, and gamma delta T cells, we get it from CD3. And then based on the specific markers, we also classify our cells and group them. The population shows that myeloid cells is reduced. Why? This we can explain based on the recruitment of these cells to the CNS in case of, as a result of MS and inflammatory response. It could be as a result of apoptosis. And as I mentioned earlier also, during the immune cell development, there might be downregulation of myeloid cells in case, especially for the MS patient who have genetic HLA-DR for that case. And iNKT cells, there is no fluctuation. You can see the flow cytometry result there that 0.03% was the population that we were able to get. So iNKT usually have stable, or it can be in pathogenesis stable or limited. That's why we cannot tell so much on the differences. Gamma delta T cells, the level of MS group decreased. Apoptosis, their recruitment into the CNS in response to the inflammatory response. So, in conclusion, the cells that we were able to study here, they had a varied distribution between the two groups. And from our finding, there were no significant differences between the two groups. And this could be attributed because of our sample size, because I've worked with 13 samples from multiple sclerosis patients and 8 LT control, which is a very small sample. And also, the samples were provided by neurological department, so information about patient history, the kind of treatment they are going through were not provided, and this will also affect the population and the phenotype of the cells. Thank you. These are the people who have really helped me throughout my project from Department of Immunology and Biotechnology, and AIBC for an opportunity to present my work. Thank you.
Thank you, Grace, for keeping time. So the next presenter is Charles Kivasu. Please note down your questions, then we'll take all the questions at the end. Okay, Charles, go ahead.
Hello, everyone. Good evening. My name is Charles Maingi from Kenya Institute of Primary Research in the Division of Evolutionary Ecology, Conservation and Ecosystem Health. And also I'm a student at Rutgers University. And my talk today is social media and monkeys, the human primate interaction in the interface, and also the narratives in Kenya. If you understand that some years back you find that monkeys were like persecuted in the media, where you find that there was a time there was a blackout, country blackout, and monkeys were accused that they are the ones which caused that. The other part is find that we had the case of M-pox, the monkey pox. So if you have been working with monkeys, you find that everywhere you go, you have been told that you are vulnerable to monkey pox. And also from the first day of this conference, you find we have seen a lot of potential zoonotic diseases, which where you find monkeys are either vectors or hosts. So from that interactions and also in that we came together and did a small study where we are presenting today preliminary data. And you find that within the human-primate interface, primates face increasing challenges. And you find that sorry, let me get this. You find that in these interactions, you find they are positive and also negative. But you find our media, personnel, and outlets, they focus mostly on the negative aspect, but not the positive aspect. And with this increased social media, you find everybody can access Facebook and YouTube, and you find that what is being presented is different narrative from what almost everybody is doing in the lab. So you find there's that disconnect between social media and what is from the lab, that is from our researches. And you find this public consumption of this information also affects conservation and also outreach, and also may also affect public health, because most of our studies are concerning on biomedical research of public health. And you find what mostly being reported is that you find in the societies that monkeys are invading farms, leading to food security issues, and also in some cases they invading houses. So you find in the study of primatology, you find that there's that missing link, and now the importance of primates in the ecosystem. And in Kenya, you find the interactions frequently feature in all media outlets, and public are now aware of the importance of primates, specifically different primates, not all primates, which have these issues. So from that, all that we came to, did a primary desktop study where we collected coverage reports on human-primate interactions through Google research, and that was our objective. And the method we did search on Google search on keywords, that is monkey, baboons, monkeys in the farm in Kenya, and monkeys, people in Kenya. And we focused on Kenya because, you know, Kenya is one of the primate range countries, and we have a lot of interaction between people and human and primates. And this was done in the past decade from 2014 to 2024. And we came up with the relevant 100 hits, and we used 50, which after filter we used 50 cases where we involved monkey and people interactions. And the data we presented, because it's a preliminary data, we presented is in descriptive and also in tables and graphs. And some of the screenshots I've presented, you find there Kenya odd power transmission still has a risk from monkeys, angry baboons, turn-on goats, and you can see, you see the narrative. The narrative is like, it's a negative. These animals are like affecting our lifestyle or our livelihoods. Then from the data, we found the media alerts, we find that the published data, mostly we find the published data nowadays are being put online, and you find Daily Nation and Kenya News and some of the online platforms are the ones leading in that. And the frequent mention of these activities were crop raiding, invasion, and snatching or feeding livestock. And one who want to focus on this is most of the of the clips featured on Kenya-based intervention and baboons, and focused on the dry season. And you find that most of these reports are generalized primates. They did not mention which primates was affecting. The other part was that they affected the staple food, that is the means, and also the beans. And different methods were being used to deter this. And also you find there was different use of mention of the names, but you find the images are different. And on the discussion is that as much is needed to report on these drivers of negative interactions. And you find there was misinformation. Like, for example, there was one clip which was showing that languas are in Kenya, but you find they are farfetch monkey. And you find these negative consumption of public sharing of information obscures research and also conservation. And more is needed in educating the public on ways of coexisting with primates. Then the question comes, so why is social media non-humans in this conference? One, because you find increased population is affecting primate habitat, and this also opens reservoirs for diseases and vectors. This affecting increases human-induced climate change, which increases interaction with primates. Also, it affects food security and also leads to one health risk of zoonotic diseases. A good example is the next clip, where I will play, but I will put a disclaimer that some of the clips, images in the clip might not be comfortable with some people. I don't know if to play. Oh, sorry. Sorry, there's no sound. So this is a clip from one of the of the TV stations, XK24 in 2008, in a place in Muranga. You find people are enjoying the mangue, and you know the risk of this can be. So I'm concluding. Sorry. So these are my references, and I want to acknowledge the division and also my co-authors and also the organizers of this event. And I want to leave you with one of the comments. If you utilize social media well, it can have a very good reflection of the society. So if you paint primates as part of our problem, they will become part of our problem. Thank you.
Thank you, Charles. Our next presenter is Winnie Kulei. Please remember to write down your questions. We'll take them after Winnie presents.
Good evening. Good evening, everyone. Okay, my name is Winnie Kulei, and I'm a PhD student at Mount Kenya University, and I'm working under Dr. Bernard Kanoi, Professor Jesse Gitaka, Professor Thumbi Mwangi, and Professor Samuel Mwalili, all of them in MKU and some in SEMA and AIMS RIC. So today I'll be presenting on assessing the effect of sailing nets in malaria control in Homa Bay County, Kenya. And I'm going to take a statistical and simulation modeling approach in this study. So, we've had an introduction of the background of malaria, and we've seen how the adverse effects of malaria. And one thing that I'm going to insist on here is that a third of population in Kenya live in high malaria risk regions. And we are going mainly to focus on Western Kenya, which is whereby malaria is endemic, and it has always shown very high entomological inoculation rate, and at the same time, there are lots of infection resurgence in the region. And so the problem overview of this study is that we know that malaria is endemic in Kenya. There are traditional methods that have been there since a long time ago, but they face a lot of limitations. And there are also new methods that have come up, and they are still understudied. And therefore, that implies that there's a need for integrated approach. And this graph shows the trend of confirmed malaria cases in Homa Bay County. In Kenya, we know that that counties have been divided into sub-counties. And here I've just highlighted 3 sub-counties in Homa Bay County, and these sub-counties shows high, they're the ones that shows high cases in malaria. And here, notably, we can see that we have Ndiwa sub-county, which has always shown, over time, high malaria cases when we look at under 5 years old and also over 5 years old. And also, so if we go deeper into Ndiwa sub-county, we can see that if we look at the trend of malaria cases and at the same time the trend of rainfall, and that also implies temperature changes in the area, we can note that there's a trend that we can see there, and that both rainfall and malaria cases exhibit strong seasonal fluctuations across years. And the peaks in rainfall will always precede the peaks in malaria cases. And normally, I mean, in most cases, we can see that there's always a 1-month lag in that. And also, there's a strong co-movement between the rainfall and the malaria trends in that. If the rainfall goes up high, then we expect that our malaria cases is going to go up high. But if we look at that graph between 2018 and 2020, we can see that both rainfall and malaria cases showed greater variability, and that could be because of maybe the introduction of LLINs. We know that in around 2018 we had mass distribution of LLINs there. We also have COVID-19, and that could be maybe that could have affected the reporting rate of the malaria cases in the region. And about 2021 going onwards, we can see that the trend came back to normal. And then, so this is just a map of Homa Bay County, and we can, Homa Bay County is the one that's shaded in darker green color. And it's about, the Lake Victoria is about 80%. The 80% of Lake Victoria is in Homa Bay County. And since we are going to look at sailing nets here, I've just shown here an illustration of how we fix sailing nets in the houses. And the sailing nets mainly help us in covering the eaves of the houses, and that is just the gap between the house roof and the walls. So it's going to cover that space there. And we know that this is the space that mainly the mosquitoes use to get inside the house and get into the human beings to get the blood meal. And so it's going to cover that, that space, and therefore the mosquitoes cannot go in. And at the same time, if the mosquitoes get in through any other space in the house, then it means that we know that mainly when they bite, they'll mainly go up the roof to take some rest after they get full. And so it means that when they go up there, they're going to be knocked down because the net is treated. And at the same time, this net is treated with PBO, and this PBO helps with the resistance that comes to the malaria. And so once it's fixed, the house looks this way, and it's actually beautiful. So, so the methodology that I'm going to use here is that it's a quasi-experimental design, and we are going to look at pre and post, and we are going to use L-traps to trap the mosquitoes there. And then we had several covariates in our model, and we fitted a GLMM with a random intercept of the cluster. And so here we are just trying to assess the probability of a light trap getting a mosquito, a female anopheles mosquito, given that it got a mosquito. And then also we looked at the number of counts of the mosquitoes that we are going to trap in the, here, and we also considered an offset for the model. And then also for the methodology, for the simulation part, we used the malaria simulation, which is online in R, and there we also looked at the climate factors there. And so for the results here, we can see that when you look at the pre-intervention and the post-intervention times, in the pre-intervention times, when you look at the mosquito counts, in both the houses that, the control and the interventions, the number of mosquitoes initially were equal, but after the post-intervention, you can see that there's a drastic reduce in the number of mosquitoes that were caught in the intervention. And then also when we look at the numbers of the houses that had 0 mosquito counts after the intervention, we can see that in the intervention region post, then we had the number of households, the proportion of households that had more, that counted more, that had more 0 mosquito counts. And then so for the statistical analysis of the probability of having a female mosquitoes, we can see here that at the post-intervention, when the post-intervention, we had a smaller probability of having a female mosquito with an odds ratio of about 28%, and therefore that means that we have a probability of, there's a likelihood of 72% of having less female mosquito in the houses that were installed with the bed nets, with the ceiling nets, sorry. And then here we are just looking at the effects of intervention and covariates in the female anopheles counts. And we can see here that the one that's statistically significant and most important is the interaction between the intervention and the post, which gave us a rate ratio of about 27. For the malaria simulation, this is how our weather elements were, that is rainfall specifically and temperature, was represented in our model. And this is the results that we got there. And we can see that whenever, if at all we have this intervention in 10 years' time in Diwa sub-county, then it means that after the 10 years, the prevalence is going to go to about 0.02, while in the final prevalence, when we don't have any intervention, it will be about 35.5%. And so I would like to acknowledge my supervisors, MKU, AIMS, RIC, and SEMA, Ministry of Health, and KMD. Thank you.
Let's give them a round of applause. So can we have Charles, Grace, to just remain there so that we can have the questions. You can join them. Grace, please join. So any questions? We have many, so.
My name is Boaz from Most, and I have a question to, I think, Grace Okicha. Thank you for that wonderful presentation. My question is, you have talked about multiple sclerosis, and you have talked about innate immunity. But I know that multiple sclerosis is a chronic autoimmune disease. And I know that innate immunity provide an immediate maximal effect. So the conundrum here is, if multiple sclerosis, if it could be chronic, I could have understand. But when it comes to time, since it is chronic, does innate immunity, I think that adaptive immunity after innate has play its role earlier. Now the adaptive immunity is the one that take role. I don't know if you have understood. You have got my question.
Could you repeat, please, because you are too close to your mic.
Okay, my question is, maybe you talk about innate immunity, and you know that innate immunity provide a maximal, it's immediate, and multiple sclerosis is chronic. I think now you could have talked about adaptive immunity, because now it being chronic, now it has switched to adaptive immunity. Or you can tell me the time that you used to conduct your research.
The time, I think for my project, the timing was between June 2024 to February 20, around like 6 to 7 months. And for the samples, we were provided from the neurological department, and the timing can differ, because sometimes we get the sample in the morning, and then the other time, depending on the comfortability of the patient also. And remember, this is a project that was done, and it's still like a study to know what is the population of these cells in different patients, which are composed of male and female. But I don't know if that's what you really wanted.
So he's asking that your focus was in innate immunity, yet she should have focused in adaptive immunity.
Yeah, because you are talking about innate cell, yeah. You talk all about innate cell. But I've told you that, you told me multiple sclerosis is chronic. Yeah. So you know that innate is the first, I think, like first line of defense. After chronic, after innate is going to shift to adaptive.
Yeah.
Yeah. But now you say that you conduct your research for 7 months. By that time, I believe that it has switched to adaptive.
Okay. I think most of the project has been done, and many investigations are mostly concentrated on the adaptive immune system. And the innate immune system was underexplored. That is why we venture into this project, and we want to see the role of this innate immune system, immune cells in the case of multiple sclerosis. In the same case, adaptive also is part of it, but my project was mainly on the innate, and for this case I mentioned is innate lymphocyte and innate T cells, which is a bridge between both both two layers of the immune system.
Okay, thank you.
So my question goes to Charles Kivasu. Yeah, thank you very much for your presentation.
Welcome.
If I got you correctly,
yes,
your research seems to paint or to show that the monkey as a victim of the bad media, the relationship between monkey and human, it's more of the human painting monkey as. So what percentage of the news outlets and the hits was wrong? Like if people say that a monkey attacked a human, was it the case, or is it because the monkey seems a victim, according to your research?
Thank you very much for your question. Actually, of the 50%, it was negative, but still we are continuing analyzing the data, so we hope at least to get at least a percentage which is positive. But currently, as you have seen, most of the media, they focus on the negative so that they can get a good content where we can attract viewers. I don't know if I have answered your question.
So you're saying more than 50% was
More than
negative?
50, yes, it's more focusing on the negative aspect.
Okay, thank you.
Welcome.
Any other question?
Okay, thank you so much. My question goes to Winnie Kulei. Did you come up with the model you're using for your simulation? And another question is it possible to incorporate other interventions and treatments in this model? Thank you.
Thank you for that question. Okay, so for the model that I used here, basically, it was a model that is already developed, just the same way that we have other models. For example, eMod. We also have open malaria. They are there. But remember, we are not. So for our case, this is what I used for this work. But we have work in progress, whereby we are trying to come up with our own model that we can use to that is mainly tailored towards our own. Let's say towards our own ecosystem, and it will be mainly on the way. The data will be, the model will be fed with data mainly from the Western region that we are mainly looking at. And so this model, I didn't develop it. I just adopted one that is there. But we have work in progress where we are coming up with our own model for that. And then, okay, for the second question. So for this, remember the model that is here, that we've used here, has already their own parameters that are there. So we are just plugging in the parameters there, and we run the model, and that is what we get for our simulation. But the model that we are going to work on, we are going to use our own, since it's tailored to our own local use. It means that all the other interventions that we look at, for example, the vaccines, the saline nets, the bed nets, and all the other interventions will just be plugged in there, and we get our own simulations.
Hi, my name is Becky, and I have two questions to Charles. So the first one is, You've said that most times the social media bring the negative part. So how did you post if they were really AI or somebody or a bot? How did you verify the posts they were really actual posts? And the second one is, did you categorize the context of the whether it was conflict or tourism or conservation based, the monkeys being affected? Did you categorize the clusters? The narrative clusters, yes.
Thank you, Becky. The first question about if it's whether AI or just created using the intelligence, most of these were like published in their websites and also uploaded in YouTube and their TV channels. So they are not AI, although we have not detected one from AI. The other second question is about categorizing the narratives. As I had mentioned earlier, that we are still analyzing the data and cleaning and coding. We are now in the stage of categorizing the narrative and under what circumstance led to that interaction. I don't know if I've answered you. Thank you, welcome.
One more last question.
Yes, for me is I'm asking the question about modeling. So before, when you chose your covariates for the models, did you do like a collinearity analysis so that you can be able to assess the effect of these different covariates with the presence or absence of those mosquitoes?
Yes, that is something that we did, and we had so many covariates, and we filtered out to the covariates that we use there for the model, the ones that I showed in the model.
I also have a question with regards to modeling. What are some of the assumptions about your model? Basically, the ceiling net as a choice of your variable, because assuming, it's like we are assuming that the house is like a closed box with zero probability of mosquitoes getting in once now you have your nets in place. So how robust is a ceiling net? Then the other one is about the other parameters within your PDE, the partial differential equation that you shared. Suppose in future you have those other parameters maybe like becoming volatile based on the time series you've chosen. Do you still believe that the simulation curve will still go down? So like also, have you done some kind of calibration for those models to make it look more realistic?
Okay, thank you for your question. So for the first question on assuming the ceiling nets, not allowing any mosquito coming in, remember these houses, it's not like the only openings that are there are the eaves of the houses. There are also other openings, and that is why at the end of the study, we had to do the light trapping of the mosquitoes that are there and do the analysis that we saw there. And we saw that we don't have like, in all of them, we don't have like zero mosquitoes coming in. We have some mosquitoes coming in, and from there, we looked at now the number of Anopheles mosquitoes that are there. So it doesn't like close all the openings. It only closes the eaves of the houses. And then for the second question on parameterization, so so for this model specifically, the parameters that we use are the parameters that are based on our data. Remember, we've done our own data collection, and therefore the parameters that we use there to calibrate our model from the ground. But the model that we are using is one that was already developed by Oxford University. And so that's why I say that we are already coming up with our own model. And in this model, we are going to factor in all the interventions that we have on the ground, the treatments that are there for malaria that are there on ground. And at the same time, for the changes in times and all that, let's say the climate and everything else, our model is going to be stochastic. And therefore, it means that it's going to capture all those differences that come in between time. Thank you.
Okay. My name is Michael Dor from Cambridge and JQuat. My question is to Grace. My friend from Must asked you about why innate. So my question is it at the onset of MS or experienced? Because when we talk about the levels of these cells were almost the same, that there was no major difference. I believe that there has to be difference because at the onset, all these cells, CD4 is trying as much as possible to recruit helper cells like macrophages, but CD8 is destroying your myelin sheath, and then natural killer cells are also trying to destroy the autoimmune cells, like even the CD8. So maybe to understand, was it at the onset of MS or experienced or over time? Thank you.
Okay, thank you for your question. Why innate? I think I mentioned something that in most of the research that are already performed and published, the cells that are mostly studied are adaptive, which are given a very, it's like given a priority in most of the cases. And then, remember, innate lymphocyte and innate like T cells, they are bridge between the two lines of defense system. And they are really underexplored. And we want to understand, maybe these cells, they have important role in pathogenesis, neuroinflammation, as a result of this progression of disease at different stages. Because for MS, we have different types, different stages. There is a relapse, there is a remitting, and sometimes there is no even going back because of the disease. So we want to understand what are the role of these innate lymphocyte, innate like T cells. Maybe they have an important role, which is maybe for further studies, even other investigators who are into immunology, they can go in and then figure out like maybe these cells are so important. That's why we use these particular cells for our study. I hope I've tried to understand your question.
Thank you to our presenters. There are many questions, but because we are out of time, we have some online presenters waiting for the next session. Thank you very much, and please let's interact with them if you have any questions. Thank you very much. So I'd like to hand over to Professor Kennedy Okeyo.
Thank you, Martin, for such a great chairing of the session. So I know everyone is getting tired, but please allow me to welcome you to the final session of the conference. And without further ado, I would go straight to the first speaker of the session. So just a brief highlights about the session. We're going to be talking about advances in precision medicine and frontier therapeutics. So the reason I introduced this is to try to highlight some of the advanced research that will lead to better treatments and also diagnostics of various challenging diseases such as cancer and other infectious diseases. So allow me to, first of all, open this session with a sponsor's talk. It's always good to appreciate some of the sponsors that do help us run the conferences. So the first talk will be, I think you can see him on the screen. It's a video recording from Toshiharu Miwa, who is the general manager at Hitachi Limited. Hitachi has been one of the most steadfast supporter of the conference. So please listen to his message, and after that, we will start the session. Thank you.
I'm Dr. Toshiharu Miwa, Managing Director of Next Research from Hitachi R&D in Japan. I'm happy to take this opportunity to represent Hitachi in extending warm greetings and wishes all attendees for 2025 International Biotechnology. I'm Dr. Toshiharu Miwa, Managing Director of Next Research from Hitachi R&D in Japan. I'm happy to take this opportunity to represent Hitachi in extending warm greetings and wishes all attendees for 2025 International Biotechnology and Biomedical Conference. I'm thrilled to learn this year's event are jointly being organized by AIBBC, Kenya Primate Research Institute, and Mount Kenya University, opening a new chapter of collaboration, which is a cornerstone of any scientific undertaking. So let me begin by congratulating the organizers of the event, in particular, Dr. Lucy Ochoa, Dr. Kennedy Okeo, and Dr. Bernard Kanoi, who have made great efforts toward the realization of this year's meeting. My congratulations also go to the General Director, KIPRE, Dr. Uedera, for his crucial leadership and support for this event, and to the Chairman of AIBBC, Professor Collins Omer, for his support. At the birthplace of AIBBC some 10 years ago, Hitachi is proud of continuing to be part of the ongoing great scientific journey to change the landscape of science and technology in Africa. We can't emphasize enough importance of science technology for Africa's socio-economic growth, and importantly, collaboration of science technology towards the realization of better health and well-being in the continent. As a technology company grounded in social innovation for humanity's well-being, Hitachi is proud of support ongoing collaborative efforts by the local and international scientific community promote capacity building and scientific growth in Africa. It's important to note Hitachi has consistently supported AIBBC activities since the organization founded in 2014. We are proud of notes AIBBC had made notable progress in creating a unique platform or share the latest science advance and cutting technologies in the important field biotechnology and biomedicine. In addition, tremendous contributions have been made towards the opening opportunity for student faculty for further their studies abroad. Indeed, more than 6 students are currently enrolled in various Japanese universities, not to mention others enrolled in various universities in Europe and the United States. Hitachi's vision is to inspire social and industrial innovation with data and technology for the people's happiness. In this regard, our company is actively involved in innovating environmentally friendly, safe, and secure social infrastructures that are resilient to the disasters. We are focused on developing cutting technologies applicable to a wide range of fields, including power systems, mobility systems, such as train and medical systems, as well as IT systems that connect them. Although our current business engagement with Africa is relatively small, we are confident that we will expand significantly towards the second half of the 21st century as Africa is projected to become a strong growth market and the engine of the global economy. In fact, it should be noted, Africa is a continent of young and brilliant people who are full of hope. Therefore, empowering Africa's young people to develop strong foundation in science and technology will be instrumental for realization the project's strong socio-economic growth. Building capacity for young people in Africa is highly critical towards creating a pool of talented, self-motivated professionals who will contribute technological advancement in the continent. In this regard, we at Hitachi are proud of say that we have hosted 2 interns from Kenya and to have established research collaboration with the University of Nairobi and Institution of Climate Research. These opportunities could not have been possible if such a scientific activity that bring people to Africa to share the research, advance network, create new collaboration. On the same note, I sincerely hope the presentation, discussion, and networking events at this year's meeting will inspire new collaboration among the participants and herald of new era of Africa growth. I sincerely hope you will enjoy the meeting and you will make the best of the opportunity to engage in the deep science discussion, share the idea, and exchange the technologies. Above all, I hope that new friendship will flourish, old collaboration strengthened, and new ones created towards a better and stronger Africa tomorrow. With those few remarks, I would like to end my address. Thank you very much.
All right, thank you very much for listening to Dr. Miwa from Hitachi. And then with that, I would like to start the session now with in-person presentations. And the first speaker we have is also from Hitachi. Ladies and gentlemen, it's my greatest pleasure to introduce my friend, and she has been with us in all the conferences we've organized, coming all the way from Japan. So please join me in welcoming Dr. Hiroko Hanzawa. She will be giving us a talk on extracellular vesicles as a potential marker for differentiation from induced pluripotent stem cells. So just a brief bio of Dr. Hanzawa as she prepares. So she joined Hitachi R&D Group in 1991. She has a PhD in biotechnology and life sciences from Tokyo University of Agriculture and Technology. That's something like JQuat here in Kenya. While at Hitachi, she has contributed to various national projects, developing interdisciplinary expertise. Her current focus is on regenerative medicine and cell therapy, working on advanced technologies such as automated cell culture and AI-driven cell design at Hitachi Kobe Laboratory in Japan. So with that introduction, please, Dr. Hanzawa, take it up.
Thank you very much, kind introduction, Kennedy. So, to organizers of this event, I appreciate the opportunity to present today. I joined AI BBC meeting in 2017 first time. At that time, the meeting room is about one-fifth smaller than that of today. So I'm really glad to find my friends in this bigger meeting rooms. So, it is, okay. So in recent years, progress has made in regenerative medicine and cell therapies for various diseases. According to this trend, it is becoming increasingly important to produce, manufacture cells and tissues for treatments while ensuring safety and efficacy. For that purpose, we now need the systems that monitor and optimize the cell manufacturing processes. But before going into details of this study, please, I would like to explain a few key points. First one is stem cells. It's okay. So first one is stem cells. Stem cells, such as induced pluripotent stem, iPS cells, are essential cell sources for manufacturing cells and tissues used for treatment as cellular medicine. Oh, I'm sorry. So, second one is that why process monitoring is important in cellular medicine manufacturing. Stem cells are differentiated into the desired cell types over several weeks or months. Currently, we assess only the final product's quality to determine whether the manufacturing was successful or not. But if it's very low or differentiation is not completed, the manufacturing batch must be discarded. This leads to higher cost, or it can delay patient treatment. It's pity. So therefore, system capable of manufacturing or evaluating and optimizing the entire processes are essential. And the third point is extracellular vesicles. Extracellular vesicles are small particles surrounded by a lipid membrane, and that release into the outside environment by mother cells. These vesicles contain cellular components like proteins, DNAs, RNAs, something like that. The phenomenon was first recognized in the 1940s as fogs around the cells, and now it is known that nearly all cell types secrete EVs. So in this study, we hypothesized that analyzing EVs present in the culture media during cell manufacturing could enable us to know the state of the cells non-invasively. So we first checked whether EVs are present in the supernatant of cultured iPS cells. So you see here, 7 days after seeding, we collected the media and analyzed them. So we observed round vesicles 15 to 100 nanometers in diameter. And so immunoblotting for exosome markers CD81 and CD9 confirmed that EVs, and specifically exosomes, were secreted into the media. Next, we differentiated iPS cells into neural progenitor cells. We corrected the culture supernatants at day 0. And in this status, cells are all iPS cells. And so after 12 days of differentiation, in this status, cells became neural lineage cells. And so comparing the microRNA expression patterns in EVs from these samples, we found that although the cell morphology barely changed at this stage, however, the contents of the EVs differed significantly. We also checked protein expression and the other components, but I saw microRNA difference is most significant, so I'm showing these data. And these tables are showing the two-fold changes upgraded mRNA or downregulated mRNAs. So then, to test our hypothesis that analyzing EVs and the culture media can reflect the cell quality, we set up an experiment. Normally, iPS cells are cultured under the standard conditions previously reported. But we modified this condition that iPS cells were cultured for 6 days in the differentiation process in FGF8 depleting media. FGF8, fibroblast growth factor 8, is involved in the neural differentiation. So under these conditions, cell morphology and the proliferation rate, light figures, remained unaffected. But differentiation was suppressed, as showing as a choline differentiation marker suppression. We then examined extracellular vesicles under these conditions. So as showing in the middle figure, immunoblotting for CD63, CD63 is a marker for exosomes, showed a significant decrease in the EV when FGF8 was omitted from media. Interestingly, we also found a positive correlation between the expression levels in choline expression in the cells at the day 12 and the amount of CD3 in the culture supernatant at day 8. So this is our key takeaways of the first part. So monitoring EV containing exosome marker were detected in the supernatants. It means iPS cells secreted exosomes. And so exosome from supernatant exhibited distinct microRNA expression patterns. And the CD3 levels in the supernatant correlated with proportion of choline positive cells, indicating the differentiation status. So, and these results suggest that by monitoring extracellular vesicle contents in the medium during differentiation, we might be able to estimate the quality of the culture at an earlier stage. So then we started new project, Raman spectroscopic measurement of extravesical fractions. Currently, EV evaluation mainly relies on staining with EV-specific antibodies. We examined whether Raman spectroscopy could assess EVs without such staining, non-invasively. We built a custom Raman measurement system, the light figures, and purified EVs from the supernatants from both iPS cells, and so after 12 days of differentiation. The spectra showed here, light form second figures is showed distinct signals around 1450 and so 28 and 2850. And so as showing the light figures, and these peaks which differ from cell debris, those of cell debris, we found to be associated with proteins and nucleic acids, and especially lipids. Then, to verify whether Raman spectra of EVs can reflect cellular changes, we designed an experiment again. This time, during iPS cell expansion, we cultured cells for 2 days without FGF2. FGF2 is a factor essential for maintaining pluripotency, while proliferation remained unchanged, as shown in the left figures. And NanoG is a pluripotency marker, expression at day 0, and choline expression. Choline is a differentiation marker. Choline expression at day 12 were reduced. This means that when the condition of iPS cells worsens, it becomes clear that subsequent differentiation is unlikely to succeed. So we set these conditions, and then we analyzed EV spectra. And so these data are showing machine learning data from EV Raman spectra. Raman spectra obtained from EVs revealed that the spectra could be distinguished based on the number of days after differentiation, showing the upper left figures. Especially day 0 and day 12 is completely segregated. And so, and also, these data are showing that and these data reflected changes in cell quality resulting from modified culture conditions. As I mentioned the previous slide, so if iPS quality is worsened, their final differentiated cells are also worsens. So these from analysis of EV Raman spectra, so we also know that result. Additionally, spectral analysis suggested changes in lipid condition. Saturated fatty acid signals were higher at D0. So upper is lower is saturated fatty acid spectra, and upper is unsaturated fatty acid spectra. So in the day 0, saturated composition of saturated fatty acid signal were higher. And while unsaturated fatty acid signals increased by day 12, by day 12 is differentiated cells. So while it is unclear why the lipid composition differs between iPS cells and differentiated neural lineage cells, but there is a possibility that physiological changes associated with the lipid are occurring in the cells. So, as shown in the previous slides, so Raman spectra of EV fraction revealed characteristic signals, especially with lipid-derived signals. And so from this analysis of these data, we also find the possibility of EV analysis of Raman spectra having a possibility to know the cellular status. So also this research on EV Raman analysis is still in the basic stage. It demonstrates that but potential, so we would like to continue this project. So today's my presentation came from collaborative work conducted with members of Hitachi and Professor Jun Takahashi team of Kyoto University and Sumitomo firm. So thank you for your attention.
All right, thank you very much, Dr. Hanzawa, for the presentation. It's very interesting that you're deploying Raman spectroscopy to analyze EVs in the supernatant. Very interesting. I will allow just one question because we are short of time. Anyone with one quick question? Somebody? All right, so I'll ask you one very quick question about, so it's interesting. You said that you see differences between iPS cells and differentiated cells in terms of the lipid composition. So do you have any theory why that would happen? Do you have any idea why the difference would come out?
Yeah, about that, so there are several lipods, but it's depend on each lipods, their idea of what's happening is different. But this is my thought. So, so to be differentiated, so the lipid layer should be soft. I don't know, just
right.
Just
It's
yes.
a theory, yes.
And so something like that, the physiological situation should be differentiated to the other cells.
Okay, very good. Once again, ladies and gentlemen, join me in appreciating Dr. Hanzawa. Thank you very much. So our next speaker is also a long-term friend, and please welcome Professor Masood from Sweden. So I will introduce you as you take the stage. Okay. All right. Just make it short because. Okay. I don't know how. Well, I will do that. So just as we wait for your presentation to be shown on the screen. So a quick one about Professor Masood. I don't need to introduce him. For those of you who have been here, you've seen him time and time. He's been coming here every year to join us for these conferences. So right now, he's the chair of the department at Uppsala University, where he is the director of clinical Proteomics and Immunology platform, the SciLife Lab in Sweden. Professor Masood has authored and co-authored multiple peer-reviewed scientific papers. I can't name them all, but I would say that his contributions are acclaimed. He's a recognized and honorable expert in the field of molecular diagnostics. Actually, he developed what we call the proximity assay, which he has been demonstrating in some of our workshops. So I don't want to spend so much time introducing his long track record, but I will allow him to share with us his insights. Thank you, Professor Masood. Please
Thank you very much. Please allow me first to thank the organization for invitation. It has been an amazing week with 3 days workshops in Tika and now 2 fantastic days with this conference. We are very late. I can give one feedback to our chairmen and women that maybe you shouldn't be so nice. So other conferences, 1 minute to the end, so the chair will stand up, and then 30 seconds before that, so they come closer, and then after that one, they stand in front of the speaker, so they don't allow you to talk so much. So maybe we should learn something from that one. Anyhow, Sweden is quite known for innovation and the tradition going back many, many years, hundreds of years, with many, especially within the life science. You know ELISA, you know protein separations, you know ultracentrifugations, and then we try to continue to with these kind of innovations. And during the years in our milieu, in a larger group, not only my group, it's molecular tools, we have developed a large number of molecular tools for analysis of DNA, RNA, and proteins, and macromolecules. But today I will focus mostly on the protein detections and the EVs. So as you can see, there are many different variants of proximity ligation and proximity extension assays that we have developed, and then the common features for these techniques that we use as many as possible probes to increase the specificity, and then connecting our affinity binders, it could be antibodies or affibodies or aptamers whatsoever, to DNA that can be amplified and also can be used for multiplexing. So these assays can be used for plasma proteomics, for single cell proteomics, for in situ analysis of proteins, detections and interactions of proteins. And also we have, since several years, we try to develop these kind of methods for detection and characterization of extracellular vesicles that you have just heard about. So when it comes to the amplification, which is very important for the sensitivity of assays, there are 2 ways to do these kind of amplifications. Either you have this regular PCR that everybody is aware of, or we can use also another amplification, which is called rolling circle amplification. It's you using circularized DNA as template, and then a specialized polymerase from 529 virus can going around this template and producing a thread of DNA, about 1,000 copies per hour, that can be hybridized to fluorophore oligos, and especially for in situ, you can see very bright signals. Just for your background information. So one of the techniques is the proximity extension assay that is developed by one of the startup companies, by Ulf Randgren in our molecular tools. And this is now available commercially and used worldwide. More than 1 million samples have been analyzed so far. There are 2 sets of that one. Either you can use it for analyzing 96 samples targeted with microfluidic PCR, or you can use the next generation sequencing, and then there is a panel of 5,400 proteins can be analyzed in few microliters. And we have people using it very widely, and we have used it in many different projects. The latest one just has been published that I'm showing here is for detection or identification of the panels of proteins as biomarkers for clear cell renal cell carcinoma, which we used oncology panels on 130 patient samples and 110 controls, and we could identify 7 biomarkers or proteins that together could give a area under curve of 1, which we had very difficult to convince our reviewers because they thought that it's too good to be true. But we did all the controls, both the technical and bioinformatical controls. Finally, they accepted, and now it's published. And at the same time, we used also regular techniques like Western blot to analyzing a set of known proteins in tissues. So we took the tumor tissues and analyzed them, and then we could see the correlation and association of these proteins to TGF-beta pathways and other cancer pathways. That this is now under continuation to doing evaluation with a larger number of patient samples. I don't need now to describe the extracellular vesicles. We had a very good introduction to that one, so it's a benefit of time, so I continue. So we have been working quite a long time now on EVs and trying to establish new assays for characterization of these extracellular vesicles, which are very heterogeneous and very complex. One of the methods that we used, of course, it was PEA or proximity extension assays that we used around 4 panels, in-house panels and commercial panels, and analyzed a large number of EVs, purified from a large number of cell lines and also body fluids like breast milk and seminal fluids. And by bioinformatic analysis, we could see that when you analyze all those proteins in these EVs, you could trace them to the original cell lines or to the organs that they are generated from. So that is a first step when we are going later into the blood to analyzing the EVs. We could easily see where they are coming from because of those heterogeneity that they have on the surface. So they have organ-specific or cell-specific proteins on the surface that we can use. So that was to see whole the proteins. Recently, when now Olink has introduced this new explorer with 5,400 proteins, we use that one, but a little differently. Now we wanted also to identify surface proteins, not only the total proteins. So in this case, we subjected both lysate and non-lysate EVs from a set of pancreatic cancers and for Olink Explore, and we could identify a large number of proteins that both on the surface and in cargo, that many of them, they are, it's nothing wrong with your eyes, I cannot even see them, those small things. So these are correlated to the pancreatic diseases and digestive proteins. So that is an ongoing EU project that we are now identifying those. So now we can go to the blood samples and trying to see the intact EVs in blood as biomarkers for pancreatic cancer. Sorry for my voice. So when you are working with affinity-based, so it's not unbiased. So another method that we have established is mass spec-based to look at all the proteins, possible proteins. And here also we were interested to see both the surface proteins and the cargo proteins. And the assay that we have established here is that we take the intact EVs, biotinylating the surface proteins, lysate them, separating the biotinylated surface proteins from the rest of the proteins, and then subject them to mass spec. And in this way, we can identify which proteins are on the surface, which are in cargo. And then we did that one for PC3, which is a prostate cancer cell lines, aggressive cell lines, and also some EVs from healthy men isolated from seminal fluids. And we could identify a few thousand, or about more than 2,000 proteins, and a few hundreds of them were not published before. So, and then we're going back and validating these surface proteins with the assays that we ourselves developed, like solid phase PLA or flow-based PLA. But here, all of those methods that I described so far in a bulk. So we are also very interested to look at the single EVs. And DU, one of my former PhD students, he developed an assay, we call it the proximity barcoding assay. It will maybe take 10 minutes to go through the details, but I very, very short, I can say that you mix your EVs or your samples, blood samples, for instance, with a set of probes that are affinity antibodies, for instance, conjugated to DNA with different barcodes. And then when you capture the EVs with those probes on it, and then mixed it with a DNA library, which has a third barcode for each complex. And then you extend the barcode on the, or the DNA on the antibody above the DNA library, and in this way you incorporate the third one in that one, and then you, after PCR and also sequencing, you can identify combinations of proteins on single EVs. So that is, I think, still a unique technique to look at EVs in this way. And if you look at 3 or 4 or 5 proteins on same, so you can separate nicely all these EVs that we had in the pool from different cell lines or from human body fluids. So these are the techniques that we have developed for characterization of EVs. And the last one, it can actually be as diagnostic also, because it's enough strong that you can see in the patient samples. There are other publications in this regard. And then now is also commercialized, and this, when we publish, it's 36 probes, but now is about 520 probes in one. So you can look at 520 proteins in one run. Once we have these informations about the cargoes, about the surface, then we can establish other assays, such as solid phase PLA, or in this case, a more complex one for PLA than that we developed a few years ago. And then you look at 4 or 5 different proteins in same antibody, and you must have all of those together in order to get a signal, PCR signal. And we have used this system for looking at the patient samples with prostate cancers, and we could see that when we divide the patients based on Gleason scores, we could see the difference between Gleason score 7, which is the real cancer, and 6, that is uncertain, and also compared to the test, the PSA test that's currently used, we could better differentiate the patient's controls in early stage. So this is one example. The other example is a flow-based technique that Lisa Love, sitting here, developed when she was my PhD student in my group. So here is, as you may know, people, they're using flow cytometers for detection of EVs, but then because EVs are very small for conventional machines or instruments, so they use a bead, and then you have many EVs on same bead. So you lose this beauty of flow cytometers that you can look at one single cell. But here what we did, or Lisa did, so we capture EVs on a capture antibody via a DNA oligo to the beads, and then we probe them with 3 different pairs of antibodies with those oligos for PLA. And then we could, after that, cut the DNA and release single EVs, and then subject them to this rolling circular amplification that I described in the earlier. And then subject, because these, then you get blobs of DNA on the EVs, some micron size, and then you can easily see it in flow cytometer. So this is an example that when we run different samples, one of them is when we spiked prostosomes from men into blood samples from women which are not supposed to have prostosomes, and we could clearly detect them. And then the microscopy images also showing that for each pair of those antibodies, we have one color. So we have 3 different colors, meaning that we have 6 different antibodies plus capturing antibodies, 7 different antibodies in order to get these results. So just to get a feeling for this flow, so if you have your EVs about 30 to 100, 150 nanometers, and you have this synchronized DNA as template that we are using for 20 nanometers, and after 1 hour, you have a blob of DNA about 1 micron with at least few hundred oligos, fluorophore oligos on that one, so they can be a large complex that is easy to see in microscope or in flow. So, and this is one short, very early test on blood samples with prostate cancer with 2 different combinations of proteins with both controls here, if somebody can see it, with only purified prostosomes, or patient samples, and then healthy controls that we could see all of it. Very few only as a proof of concept that ASN has run that one. And now we are starting to analyzing biobanking materials on that one. So with that one, I would change the year and talking about something else. During the COVID time, so also our lab starting to looking into the detecting antibodies instead of proteins. And that was the lead, project leader for that one is Ulf Landegren. I was collaborating here. And what we did here, that we changed the idea that using antibodies to detecting proteins, and here we took the target proteins and labeled with DNA to detecting the antibodies. And in this case, each line is 1 patient with COVID samples, and the others in the bottom, they are controls. So we could see the dilution here for patient samples that we got to look at the anti-S1 antibodies or anti-NP antibodies in the patient's blood samples. So they have been infected, and then there is antibodies. And another way to use these systems also, we established 3 different assays to detecting different IgG, A, M, and IgG, or IG I meant. And then following after the vaccination, so you can see the vaccination started the week 1st, and then boosting second vaccination was after a few weeks, so you can follow for each kind of antibodies, and the total also total antibody against S1, so you can see all of those and can following how it increase or then decrease also. And the last one is actually not protein. So some of you already heard that one in the parallel session before lunch. That's another, during the pandemic, we established the assay to detecting COVID viruses. The idea was to beginning to see those in flow or by other methods, but actually it was a failure because it was very difficult, but then we managed to convert that failure to success, and we could develop this assay with some new idea on padlock to see the virus genome without any necessary of any instrument, and using this rolling circle amplification on the DNA. And now there is a commercial company around it. Lisa is CEO for that one, and then the idea is to launch for the first products, two different, one for virus and one for bacteria, but both in multiplex. So maybe you use for viruses, tell you it's not COVID, but it is influenza A, for instance. So that is ongoing, and then, yeah, it's commercially available soon, hopefully. So with that one, I thank you all contributed to these results, and for your attention so late in this afternoon. Thank you.
All right, thank you very much, Professor Masood, for the exciting talk about your technologies. They are very deep. So in the interest of time, I would like to invite one question from the audience. Anyone with a question? We are running late. Yes. I will jump and give you the mic.
Thank you, Professor Masood, for the presentation. I just saw something very interesting in your amplification method, the rolling circle amplification. So with the products you're having after the amplification, they form something that resembles the DNA network. So is it possible to visualize these via naked eye? Because you mentioned you can use a microscope and flow cytometry. But would it be possible to visualize these via naked eye without the need of microscope? And maybe how long would it take to perform the amplification?
With naked eyes, I thought the last image was that one. So you use those padlocks, several of those, on the genome, and then you do this rolling signal amplification, producing a lot of those, and then because you put it on the beads, some kind of technical that I didn't want to go into because of the time, so then it will be aggregated, and then it is what you see it now with the naked eyes in the tube. It's what I showed here. That's yes.
Oh, so how long would it take?
30 minutes in this assay that Lisa has in the company, right?
Okay, thank you.
Thank you.
Thank you so much for the great presentation. Just a quick one. Are you able to use samples that are obtained using non-invasive methods like saliva, urine, especially working with the EVs? Can you use urine or saliva as samples for
it
your
is
assays?
shown that this, EVs are present in all kind of samples. We have done it for few different, but we have an ongoing project for oral cancer. There we are looking into that one for saliva to get to find EVs as biomarkers for oral cancer. Yeah, that is possible, yeah.
All right, so ladies and gentlemen, please join me in appreciating Professor Masood. Thank you very much. And, all right, so our next speaker, all right, I'll keep it brief. Okay, so please allow me to welcome Professor Jakob. He's been with us also in the past, leading bioinformatics and the genomics course in the workshops. So Professor Jakob is a professor of bioinformatics at the Department of Molecular Medicine and at the Bioinformatics Research Center, Aarhus University, Denmark. I hope I got that right. His field of research is computational genomics with interests both in method development and large-scale data analysis. He has numerous contributions, and I will not read them all, but he has a huge interest in comparative genomics, RNA structure predictions, RNA biology, ancient DNA, cancer genomics. Take it up and share with us your insights, please. Thank you.
Yeah. So thank you so much, Kennedy, and thanks to all the organizers for inviting me once again. I was here 2 years ago, and this time around again, it's a huge pleasure to be here and to get the energy from all the students and see the exciting science going on here. So I'm going to talk about be giving an introduction to somatic evolution, and then talking about a study we've done on the effects of genotoxic chemotherapy on healthy cells. And the work I'll be presenting was headed by Gustav, who's a postdoc, who's currently a postdoc in my group. So the majority of the cancer chemotherapies that are given are genotoxic, which means that they work by introducing a catastrophic amount of mutations in cancer cells, which lead to the death of the cancer cells. Can you hear me okay? Yeah. However, we know that chemotherapy also introduces mutations in healthy cells. The chemotherapy is usually given systemically. It has a chance of affecting all cells. And we know that, we probably all know that people often lose their hair because cells in the hair follicles die. And people may also experience neutropenia with a decreased amount of neutrophils. So fastly dividing cells are certainly affected by this. However, we know much less about the long-term effects of chemotherapy and whether they actually affect stem cells and whether mutations are accumulated in stem cells that then lead to a higher mutation burden in cells in the body in general. We have therefore conducted this study with a focus on hematopoietic cells. However, to get to the study, I would first like to introduce several necessary backgrounds to understand what we're doing. So first I'll give an introduction to what somatic mutations are and to somatic evolution, which I think is a really exciting topic. Then I'll introduce the concepts of mutational signatures, and finally circulating cell-free and tumor DNA. So replication is not perfect. So for every mitotic division in our body, 1 to 2 mutations accumulate. This means that during cell differentiation, more and more mutations will accumulate in the cells of the body. The range here goes from tens of mutations in germ cells, so in gametes, which is the lowest, up to thousands of mutations. So another important concept here is that we may have mutations that hit cancer driver genes and which change the proliferation rate or the proliferation capacity of these cells, which can lead to rapid expansions, and we call these clonal expansions. So we have a whole groups of cells which derive from the same cell at some point. So a brief introduction to cancer development here and to somatic evolution as well. Cancer progression, or cancer is really a genetic disease, and this is a schematic of cancer progression, starting from the zygote, where we have no mutations, which defines the germline for the individual. And then we are here looking at the accumulation of mutations in a small number of key cancer genes. And most of the mutations that accumulate will be so-called passenger mutations that do not affect the phenotypes of these cells. However, some mutations, I can't really point to them, but denoted by these other types of symbols, small wheels, they will give the cells proliferative capabilities and may, for instance, lead to these early clonal expansions that are they are not tumors, they are early neoplasms. As more and more of these tumor mutations accumulate, we may get invasive cancers, and during chemotherapy, we can get a large accumulation of additional mutations. So the focus in this talk, that's not how it looked in my slides, but anyway, the focus in this talk will be on somatic evolution, so up to the formation of tumors. So we haven't been able to study somatic evolution until recently, until the advent of high-throughput sequencing. And this study by Inigo Martinez-Zero in 2015 really pioneered this. So what Inigo did and his colleagues was to obtain some skin tissue from individuals that had gone through cosmetic operations for hanging eyelids. And they then sampled really small areas of these eyelids and subjected them to deep sequencing. By doing that, they could evaluate the mutational frequencies in these small areas, and they found that there were a lot of clones, some of them covering several square millimeters, and they could also define the phylogenetic tree or the little evolutionary tree relating the cells in these clones, as shown here. And they have done all these nice visualizations of the clonal composition of these tissue, of skin in this case. And you can see that in this individual, really the majority of the skin cells come from clonal expansions. So meaning that that individual, but also all of us, are actually full of these you could call them early cancer stages, where the cells have accumulated several driver genes and started to expand, but it's still completely benign. Another study done a few years later was on the esophagus epithelium, and there they had a range of age, or a wide age range included among the individuals screened, and they found that, or you can see here the range from a young individual to a somewhat old individual. You can see that both the size of the clones and the amount of mutations really increase with age. So, the types of tissues that accumulate the most mutations are the ones exposed to the environment in one way or the other. So the outside environment can be harsh, and there are mutagens, for instance, UV light that introduces mutations very efficiently, and different mutagens often have different mutational preferences for different contexts. So this is captured by these so-called mutational signatures that denote the fraction of mutations that are expected in different sequence contexts along the genome. So for instance, UV light has a preference for creating mutations at dipyrimidines and creating C2T mutations. And all of these, we now have a lot of knowledge of the mutational signatures corresponding to different mutational processes. And these are cataloged, and they're given names like SPS7A, which is single base substitution 7A, which corresponds to this UV light signature. So tobacco smoke is another very potent mutagen, which primarily creates mutations in C2A contexts. And then these genotoxic chemotherapies like 5-FU are, of course, also potent mutagens, and 5-FU creates these types of mutations, primarily of the T2G type in certain contexts. However, for a given individual, we do not observe the result of these mutagens in isolation. Rather, we observe the accumulated outcome of all of them. However, we can decompose this distribution of mutations now observed from a tissue sample from some individual into its different constituting processes. And for instance, if in a skin tissue sample, it would have a very high chance of primarily being UV light that caused the mutations, and for a smoker, a lung tissue sample would be dominated by tobacco smoke mutations. It's not only single base substitutions that are cataloged, double base substitutions are as well. And for instance, platinum chemotherapy gives rise to these very distinct double substitution patterns in the genome, such as this CT to AA mutation, which occurs at a more than 40% rate for this type of mutagen. Then a brief introduction to circulating cell-free DNA and circulating tumor DNA. So in cancer patients with solid tumors, the cells of the tumor will shed DNA into its environment when the cells die. Some of this DNA will make its way into the bloodstream, where it floats around in the plasma, together with circulating cell-free DNA from healthy cells, which really dominate the DNA there in the plasma. We can spin down the blood cells and purify the DNA from the plasma and sequence that. One of the reasons we're interested in this is that ctDNA rises with the tumor burden during cancer development. So in early stages of cancer development, while the tumor is small, there'll be really minute amounts. But as the tumor grows, the ctDNA fraction grows. And in this little schematic, we imagine that the tumor is onset out at the left end, and then at some point symptoms arises, and the cancer is diagnosed, and hopefully the patient will have the chance of an operation with curative intent. However, these operations are not always successful, and sometimes there are some cancer cells left. So we know that, for instance, for colorectal cancer in Denmark, more than 30% of the patients have recurrence after surgery. And this ctDNA monitoring gives us a chance to identify patients with residual disease really early. Like in our case, it's a year earlier than imaging techniques can do. So usually adjuvant chemotherapy is given to eradicate this residual disease. However, still 20–30% of patients have recurrence. Again, ctDNA gives us a chance to monitor the tumor development, and however, fewer are cured in the end, even though they may be given additional chemotherapy. Here, ctDNA may give us a chance to guide therapy to see whether it's working or not, so to monitor the response and to survey the presence of resistant clones growing up. And then finally, in the ctDNA field, the holy grail is really to be able to detect cancer pre-symptomatically, so earlier than we're doing today through population screens, which would really increase our chance of curing patients. So the earlier you detect cancer, the higher chance you have of curing the patient. So this finally brings me to our study. So here we are exploiting that we have some really extensive data sets locally generated by Claus Lindbjerg Andersen, a close colleague, on whole genome sequenced cfDNA samples. So we have this longitudinal sampling of cfDNA for patients, for colorectal cancer patients before and after surgery up to 3 years. We have a total of more than 1,200 samples. I should say that 25% of these relapse, the rest do not. And as I indicated before, the majority of the cfDNA come from healthy cells, so 85% are bone marrow derived, with the ones that are ctDNA positive in average just having 0.2% tumor DNA. This gives us the chance to study somatic mutations as well as the cancer mutations by looking at this DNA. We are exploiting that we have sequencing of short fragments, and therefore we have overlapping reads that gives us a chance to identify, to weed out sequencing errors by identify consensuses between the two reads and get high quality mismatch calls. So this is the type of data we end up with mismatch calls per patient, and we count up their distribution across the different mutation types that I mentioned previously, and then we decompose these mutation or mismatch profiles into the constituent mutational signatures. When doing a linear model analysis of this, we can evaluate which signatures and hence which mutagens have contributed to these mutations. And in line with expectations, we find that the patients that are ctDNA positive, so the ones that have tumor still in the body and are exposed have been exposed to therapy, they have significant contributions of 5-FU and these platinum genotoxic chemotherapies, and we can actually estimate the mutation rate in the tumors, and down here you can see the mutation rate in mismatches per megabase. But more interesting and closer to the real goal of our study was to see what it looked like in healthy patients. And we do see much smaller, though. We see an effect of the genotoxic chemotherapies in healthy patients. However, it's much smaller than for the cancer patients, though significant. Finally, we find these interesting signatures from the ApoBec system, which is an indication of replication stress in these cells, which have also been seen in cell lines previously, and we see this in healthy cells from these patients. So it seems like the cells and probably the stem cells are exposed to some replication stress that leads to mutations. When we then compare the excess of mutations in therapy-exposed patients versus the ones that are not, across the time series, we get this picture with not a significant difference to begin with, but after 6 months when the chemotherapy was administered, we start to get a significant excess of mutations. And the same for a double base substitution signature. So in conclusion, ctDNA shows a clear signal of chemotherapy-induced mutations. Circulating free DNA from healthy cells show weak but significant chemotherapy mutation signals, and replication stress mutation signals are higher and increase after chemotherapy. So I'd just like to thank my co-authors and the funding agencies. And I would be happy to take questions if you have any.
Right, thank you very much, Professor Jakob, for the exciting talk covering even circulating tumor DNA. We are running out of time, seriously. The food is already getting cold. One quick question, please, and we can, okay. Let it be quick.
I'd like to know that which gene specifically was mutated most when it comes to chemotherapy, and secondly, you say that circulating tumor DNA can be used in monitoring cancer, and we know that there is always DNA enzyme in blood. Can that affect the efficacy or the sensitivity of that test?
So I think I need you to repeat the last question, but I'll first address the first one. So you are asking whether we are seeing specific genes being hit, and that has not been our focus. So we are looking at whether there's any signal of these mutations in our data. And given that the protein coding regions of the human, of genes in the human genome only span a little more than 1%, 1.2%, the vast majority of the mutations would fall outside of these protein coding regions and away from the genes in general. So we don't have that much power to address the question you had there. But actually, I didn't get everything in the second question. but maybe we talk about it
Right, you can
afterwards.
have some discussion.
Yeah.
We have
Fine.
Right. The party will give you some more power to do that. So please appreciate Professor Jakob. And well, last but not least, the final speaker of the session and I think of the conference will be Professor Douglas Perkins. He's joining us online, so please just set him going as I introduce him. Well, perhaps he's known to all of us, and Professor Douglas has been working with many institutions in the country, Maseno University plus several others. I think he's a good friend of Professor Collins. Well, he leads the longest running continuously funded global infectious diseases training program sponsored by NIH, the Fogarty International Center. He has trained over 100 long-term trainees across the US, Kenya, Ghana, Thailand, South America. His research primarily focuses on the human host immune response to parasites and viruses. He has published so many papers, I will not name them all, but he has made tremendous contributions to SARS-CoV-2 detection, and also he's made significant research contributions to this field. So I will just head on to welcome Professor Perkins, please. Could you take it up?
Hi, can you hear me, sir?
I can hear you loud and clear, thank you.
Wonderful. I really appreciate the invitation. I'm sorry I could not be there in person again this year. I was on an NIH study section, but as some of you know, our government is closed, so I did not end up going there. But in any event, I thank you for the invitation. I'd like to give a special shout out to my friend and colleague for the last 24 years, Professor Collins Uma. And greetings from New Mexico to you all. I am here with Shirley Wasena, Dr. Clinton Onyango, Dr. Heidi Hurwitz, Dr. Kuyun Ching, and Dr. Kristen Schneider. And so it's really late in the day, and the only thing keeping me and you all from your food and alcohol is this lecture. So I thank you in advance, and I ask you to pay attention over the next 2 hours of this lecture that I will give. That was a joke. I'm waiting on you to laugh. So let's go ahead and start, folks. Sorry it's late for you. So I want to talk a little bit about sickle cell anemia and severe malarial anemia, two overlapping very severe disease sequelae. This will be a continuation of what Shamim Osada, who's a PhD student in the group, presented earlier. I'll just be as brief as I can, given that she's already provided some background. So if we just think about malaria endemicity in the presence of sickle cell anemia, which is a beta globin mutation, there's a tremendous overlap. That is a polymorphic variability that's driven by susceptibility to malaria. And perhaps the most important, based on its clinical features and what it defines, polymorphic variant in the human system, okay? So there's tremendous overlap. So sickle cell anemia, 80% of the global burden of sickle cell anemia is actually in sub-Saharan Africa. That accounts for about 150, 15,000, depends on where you read, but it's up to 300,000 deaths. That represents 5 to 10% of all the deaths in pediatric populations under 5. So it's a giant, giant problem, particularly in sub-Saharan Africa. And so the malaria mortality has already been talked about by others. In sub-Saharan Africa, it's 76% of the children under 5. Severe malarial anemia, just let me touch on that. In this area in Western Kenya, where we've worked for the last 24 years, the primary manifestation of severe malaria is, in fact, severe malarial anemia. And we do not see cerebral malaria in that region, which is typical of many other hypo or hyper-endemic and holoendemic malaria transmission regions, right? So sickle cell anemia increases susceptibility to anemia in general. I'll show you some measurements in 1,654 children with greater than 19,000 visits, and that will drive that home for you. And so when we talk about therapeutic discovery, one thing to think about is adjunctive therapies in addition to what's currently going on to mitigate some of the problems that we see are a little more, let's say, chronic immune activation in these individuals. And so I'll quickly go through this. Shamim presented earlier some of the pathophysiology of sickle cell anemia. Let's just highlight a few of them. We oftentimes will see children, it's very sad, who at extremely young ages have had multiple strokes, and it's very, very debilitating. About 3 weeks ago, we were working with a child that had 4 strokes before the age of 4 years old. So stroke is a very big problem. You have these vascular occlusions that take place. There's an acute chest syndrome, which is also clinically a very severe problem in these individuals. And so one thing to think about when you see these children, for those of you that do more clinical medicine, you will vouch for the fact that these poor kids are in excruciating pain from these occlusive crises that they receive. So there's also a papillary necrosis, which is essentially the sickled red blood cells at the papillae of the renal tubules, literally clogging that and causing acute kidney injury. Okay, hallmark physical sign is you see these really swollen hands and feet, dactylitis. Splenomegaly is a common problem. So when those sickle cells are being removed from circulation, they're essentially filtered out by the spleen. And then another hallmark clinical sign, of course, is the jaundice, and that comes from the hemolysis that occurs in those children. So that's sickle cell anemia, and in its most severe state during a sickling crisis. Then we have severe malarial anemia, which you all know is overlapping, right? So that can be defined as hemoglobin less than 6 based on a population norm in males and females under 5 years of age in Western Kenya. That's actually responsible, that particular sequelae of anemia in the context of falciparum malaria is responsible for the greatest amount of malaria-associated mortality worldwide. So the mortality in holoendemic regions, such as where we work in the CIA community, can be up to 30%. So here's an interesting factoid. Greater than 50% of the mortality occurs within the first 24 hours of hospital admission. The reason for that is the children become listless, become anemic. It's not an overt clinical sequelae. Mom brings them to the hospital late, so by the time we see them, they're often in an advanced state. As I mentioned, cerebral malaria in the region where we work is extremely rare, doesn't occur. It's actually meningitis most of the time. And so the pathogenesis of SMA, if you look at the number of parasitized red blood cells, it's disproportionate. So in other words, in children with severe malarial anemia, they will actually have lower levels of parasitemia. And the reason for that is they are immune naive, and so they have an overzealous immune response immunologically to the parasitized red blood cells, okay? And then ultimately, your hemoglobin concentration is going to be a balance of both hemolytic destruction within circulation as well as inefficient erythropoiesis. And so I just wanted to bring it to reality. I know it's late in the day, but some of you might appreciate this. This is some of our work in Western Kenya. These pictures, I think, are from a couple months ago. So this is a picture I have. This is Shamim here in the mask. She's an individual whose data I will be presenting today, along with a 20-year composite of data that this talk really encompasses. And this is on one of our R01s that's looking at a multi-omics approach. This is Dr. Sami Anyona. And so one of the things that's a challenge is early diagnosis. So in most of Kenya, they'll use a sickling test, and the sickling test, unfortunately, isn't very accurate. And much to most people's surprise, when you're carrying that protective allele, i.e. AS, because there's the S allele, the sickling test will sometimes show up positive. So one needs hemoglobin electrophoresis and or some more sophisticated methods to do that. So one of the things we do is we work with the folks at the hospital and the community. And so we provide hemoglobin electrophoresis testing so that we can definitively say which children are actually carrying the disease. There's another challenge with infection prevention. So some of you may know that children with sickle cell anemia are put on an antibiotic as well as an anti-malarial, okay? You also want to make sure that they're appropriately vaccinated against pneumonia, Haemophilus influenza B, and Neisseria meningitis. That's what those three are. And so the prevention scheme for malaria is insecticide-treated nets and daily proguanil. The problem with proguanil or paludrin used as a monodrug is that there's a high level of resistance. If that wasn't a large enough problem, there's also an enormous supply chain management issue in Kenya, such as they're not taking that particular medication because it's not available in the market space. So we have switched over to using artemether and lumefantrine one time per month in the children parasites, positive or not, and that's been preventing some of the sickling crisis in the population. So other things that would be required, of course, were folic acid. The children need to maintain hydrated, and so we train moms and caregivers on how to do those particular things. The current therapy is 6-hydroxyurea, which some of you know is a chemotherapeutic agent, quite old, one of the only medications that's available in Kenya for the treatment of sickle cell disease. It also requires CBC monitoring and close following because it can lead to a neutropenia. And then, of course, during an acute crisis, the children come to the hospital. You need to do pain management for the vaso-occlusion, have an available blood supply, and try to get those children back into a state of non-profound severe anemia. So this particular composite here, left and right, is a cross-sectional depiction on the left, and on the right, this is a longitudinal depiction. And so this is a study that we performed in 1,654 children. We enrolled them between 0 and 1.5 years of age, and then once we enrolled them, we followed them for 36 months after that. And that accumulated to one of the largest cohorts that's been put together in sub-Saharan Africa on severe malarial anemia. That amounted to 19,893 visits. And the reason I just want to illustrate that number is when you get that many people, you certainly, and that many visits, you certainly have robust data. And so if we look here on admission, this is, a rain cloud plot, it's called. All you really need to pay attention to is these are HBSS carriers, i.e. children with sickle cell anemia. So when they come in and we see them for the first time and then start following them for 36 months, they come in with a lower hemoglobin level. The one thing I want you to pay attention to on this particular panel here, this is the longitudinal trajectory of hemoglobin across time. Let's focus on this line right here in red. The real take-home message of this is children who are carriers of this SS are walking around in the environment most of the time in a moderate anemia state. When they get an additional anemia promoting event, such as a bacterial infection, a viral infection, malaria, it pushes this down below the threshold. And so you can easily see why they're on the cusp all of the time across that first phase of life to developing severe disease. And so this is a fragility model that I want to define for you. And I don't want to belabor it too much. I just want to show you the high points in the interest of time. So we'll look at those children and we'll say across that 36 months follow-up, how many developed malaria, how many developed severe malarial anemia, and a frailty model was used to calculate the hazards. There are covariates in the equation to account for other things that could be contributing to the anemia and or the malaria across time. So the take-home message really is that sickle cell trait, i.e. 1A and 1S, that's actually protective longitudinally against malaria. Here's a surprise to most people. When you're carrying two of these sickling alleles, you're also protected against malaria. So you might ask, well, why is it that those children have much more severe malarial anemia when you come over here and compare the two? The reason why is that albeit they get infected less with malaria, there's some inherent protection by carrying two sickle alleles. When they actually acquire the parasite, they have a catastrophic sickling event. And so you can see there's a profound increase in the risk here. This is a population of children that also has HIV. In the interest of time, we won't talk about that too much because I know you all want to go have some drinks and some dinner. But let's just say that HIV infection and HIV exposure. The difference between the two, these are children that are carrying antibodies from mom, i.e. mom had HIV positive, but they definitively did not have detectable virus, HIV virus in the system. Whereas the infected children, this is confirmed on 2 consecutive PCR reactions, definitively finding virus in the child's system. So they grew up in an environment, we're lucky enough not to acquire HIV, but grew up in a, shall we say, a challenged in utero environment, and these kids definitively have it. So the bottom line is, when you have HIV infection, you're protected because your immune system, you look very much like these children with sickle cell anemia. Your immune system is ramped up until you get to that catastrophic state. And when you actually acquire malaria, you crash with your hemoglobin level just as the children with sickle cell trait do, okay? In the interest of time, I won't read all of this. I'll simply tell you, so those are children looking to see whether or not they have altered susceptibility to malaria and the primary clinical outcome, which is severe malarial anemia in the region. Now, if you take aparasitemic children, i.e. no malaria on board, and you just look at severe anemia, kids that are carrying these 2 sickle alleles also have an increase in severe anemia. So during infections and without infections when they're walking around in an environment theoretically healthy. This is the big one, mortality. Interestingly, despite this protecting against malaria, severe malarial anemia, the sickle cell trait does not significantly protect against death. However, those kids with sickle cell anemia have a profound increase in mortality. And so in the interest of time, again, I know you guys want to have some drinks and some dinner. I won't go into this model. Let's just hit some high points. There is therapy for liver stage disease, i.e. primaquine, right, to eradicate the parasite within the hepatic cells. This is the phase in which we become clinically symptomatic. There are many blood stage chemotherapies. That's how we currently deal with the disease. When you give compounds, is this animated? The animation's not working, I'm sorry. But in any event, you have a hemolysis when you put antimalarials on board because it ruptures the red blood cells. So if you have a child with parasitemia and a low level of hemoglobin, you need to group and cross-match that individual because when you put your antimalarial on board, you're going to cause further hemolysis. You're going to see a rapid drop in hemoglobin. So really the money shot is down here in the lower right panel. This is an in vitro erythropoietic model that we've developed because as you can imagine, it's difficult to get access to bone marrow in these children. But suffice it to say, this peripheral inflammatory response that occurs in children, those signals leach into bone marrow and impact on hematopoietic progenitors and how rapidly one can make new red blood cells in the face of that pathophysiological challenge from malaria. There are no therapies that are immunological and or that address these bone marrow derangements, which are a major pathological feature of the disease. Sorry, the animation is not working. So I'll quickly talk about some of our multi-omics approach for identifying therapeutic targets in the spirit of therapeutic discovery. I'll hit the high points. We look at DNA variation. We initially did it through a 2.44 million mark array. Now we're doing sequencing. We supplement that with validation for some of the main SNPs. It's a custom design Affymetrix 200,000 SNP chip that we use. The RNA-seq that I'll present today is done by Novogene, and it's essentially a 3' ligation to the library. So you amplify all expressed transcripts within the human system, and you get between 54 and 57,000 transcripts. That can be validated by either PCR arrays, or oftentimes we'll use an Illumina bead chip array that detects 47,000 transcripts. I won't have time to go in this today, but in the spirit of our friend from Sweden, there's an aptamer-based technology from a company in Colorado known as SomaLogic, and it's been a breakthrough in proteomics. And so in 55 microliters of blood, we can get around 7,000 proteins. The technology just improved, and we're running assays right now. So in 55 microliters of blood, we can get 11,000 plasma proteins. So you'll take the plasma proteins and you'll look at the transcripts, try to find agreement on directionality that matches temporally, and then create some therapeutic targets from there. And I'll just give you a brief overview. The reason I put this in there is these are children with sickle cell anemia. These are children with non-sickle cell anemia, i.e. these are children with wild-type allele HBAA and HBAS lumped together in that category. So you have 7 children here. All of these children, i.e. the ones with HBSS, which are 7 and not carrying those 2 alleles, are 18. All of these children, however, have severe malarial anemia. So if you want to look at the impact of sickle cell disease, you have to also make sure you're in the appropriate phase of disease. So this is the worst of the worst, i.e. the lowest hemoglobin levels. It's not presented here, but the level of hemoglobin in the children with sickle cell anemia, the median was 4.1, and it was 4.3 in the children with severe malarial anemia not carrying the 2 cyclin alleles. So from that group of individuals, we got 2,820 differentially expressed genes that were FDR corrected, 2,110 upregulated, 710 downregulated. So if you go to cluster 1, this is downregulated. You can see this here. These are the 2 upregulated clusters. And so the information that Shamim presented earlier was going into those clusters and then doing additional modeling to see which genes are there, which genes are potential therapeutic targets, etc. This complicated panel on the right, I won't go through in detail. What these red thermometers indicate is upregulation, and the height of their upregulation is based on a log-based 2-fold change. And so essentially, transferrin receptor 1 is upregulated in this context. GLPA is upregulated as well. So this is a model that's annotated based on relationships of these different genes within the pathway. And so what the thermometers are showing is this exact group of children, what's upregulated, what's downregulated. The reason I want to show you this is the top emergent pathway in the bioinformatics pipeline is actually vaso-occlusion, okay? And that makes sense given the fact that this is anchoring on specifically what do children with sickle cell anemia have as unique features when they have severe malarial anemia versus non-carriers, okay? And so what you would do, for example, is you would find genes that one believes are critical. You can do an overlay in this informatics pathway, and then a list of drugs will come up that modify the actions, for example, of TFR1. So I don't have time to go through that discovery process in the time we're allotted today, but that's the next phase, okay? So I want to skip and say there's hundreds of drugs, not all of which are FDA approved. We've gone through over the past from 2014 onward, this was published in 2016, and tried to find therapeutic targets that would be robust and could target multiple pathways. One of those compounds essentially that we don't normally consider a drug is L-glutamine, okay? So what we did is we went into children, this is about 120 children stratified into 2 groups. So children that do not have severe malarial anemia, right, but a comparable level of parasitemia, have higher circulating glutamine levels than the children with SMA. So in other words, this non-essential amino acid during the infection process per se is decreased in children with severe malarial anemia. Again, I don't want to go into granularity, but this is a simple correlation saying your glutamine level is tightly correlated, somewhat tightly correlated with hemoglobin. Overlaid here is, this is 420 millimolars, and this is 2 standard deviations below the median. All this is showing is that when you have low levels of glutamine, it is a strong predictor of developing severe malarial anemia. In fact, one of the strongest predictors we've ever found, and we've looked at thousands of different genes. So low glutamine is a strong predictor of the development of severe malarial anemia, okay? So what is glutamine doing? Well, let's hit the high points. It's non-essential. When you come under a state of pathophysiologic stress, it becomes essential. It primarily comes from skeletal muscle. It's a precursor for arginine and glutathione, and that makes it important. Here's glutathione because it provides an antioxidant role, okay? It also supports red blood cell membrane integrity and antioxidant defense. And then that pathway that I just showed you, glutamine has the ability to decrease oxidative stress, thereby stabilizing endothelial function, downregulation of those receptors, decreasing the vasoocclusion. So the bottom line is here we have this centrally, you're going back and forth with glutamate, you're developing a trimer for glutathione, providing anticellular stress. Glutamine also works through an important pathway that we don't have time to talk about today, upregulates something known as heat shock proteins that stabilize intracellular protein and trafficking, therefore protecting against stress. We won't go into the autophagy method, but anyways, it can block some of the autophagy. It's also required for proper immune function, okay? So serendipitously, there is a company in the US known as Indare, okay? So they developed a compound and people were curious what it was. Indare is literally L-glutamine, okay? Now, L-glutamine therapy, IL Indare in the US is $32,341 a year, making it completely unaffordable for the rest of the world. The good news is Indare is L-glutamine. All they've done is used a slightly different modification so that they could get rights over that particular compound they made, but trust me, it's L-glutamine. And so there were 2 clinical trials that set that up. So the bottom line is when you were given Indare, i.e. L-glutamine, you had a decrease in your crisis rate.
Hello,
okay?
Prof.
In these 2 studies.
We are running out of time. I'm getting a notice. We have to stop. Please wind up.
Okay.
Thank you.
All right. Maybe 2 years from now when I'm there in person, y'all can keep time better. So the summary is hemoglobin trajectories are altered, right? Glutamine is a good therapy. Thanks to all these people, and we're great for the money that we receive. Have a good evening. Enjoy your drinks. Have one on me. Thank you.
Thank you. Thank you. All right. Thank you very much. So we are out of time. Very, very, very much. Any quick question? One, and we are gone. Okay. There's one quick question there. I don't know whether the mic, please, over there, over there. Are you there still, Prof?
Yes, I'm here.
Douglas is gone, I guess. Huh?
No, I'm here.
Oh, you're here?
Yes, sir.
All right, thank you.
Hello,
Of course.
Professor. Thank you for your presentation. I had a question about common sort of treatment approaches that are utilized in sickle cell and how that could affect someone's vulnerability to acquiring malaria. So I was curious about splenectomy in general. Do you see that potentially children who've had complete or partial splenectomy, are they at higher risk of acquiring severe malaria?
Yeah, the one thing, it's an excellent question. Thank you for asking. We do not do a lot of splenectomies in children in Kenya. We dodged a bullet recently. We transfused a child with sickle cell anemia 8 times. We almost lost him. His hemoglobin level went to 2.1. It was probably more of an act of God than our genius. We ran every single test we could. And the answer to your question is there are not a lot of splenectomies done in that reason for exactly the question you asked, which is you're creating some susceptibilities to the development of severe malarial anemia, right? And so it's a trade-off between how destructive has it become with this hypersplenism, because you have these really highly hyperphagic splenic macrophages. So when you transfuse a child, once you get that hypersplenomegaly and the overactivation of the immune system, when you put cells in it, literally, you'll see hemoglobins drop within 4 hours, 5 or 6 points. And if you think about what I just said, if the kid's at 2.1, you don't have that. So you get the blood on board, comes up, and then you'll see a rapid drop. And so it's a challenge to manage sickle cell anemia in a holoendemic region, because unless it's life-threatening and the child cannot stabilize hemoglobin at all, you wouldn't do a splenectomy, and only in really rare cases. Great question.
Thank you.
You're welcome.
Right, so with that, we close the session, and once again, let's applaud all the speakers of the session. Thank you very much for your patience. So now comes the big time. The party will start soon. Dinner will be served outside, so please, you will walk outside, pick up your meals, and come inside. We will have a closing ceremony as we have our dinner. Presentations, you should expect something, so we will all proceed after this. Let's get our meals, come back. The rest will proceed as we eat. Thank you.
Thank you, Kennedy.